Welcome, Swagatam, Suswagatam, Suswagatam. I request all the dignitaries on the dais to offer floral tribute to the idols of Padma in 1920 and since then touched many lives of downtrodden as well. Dignitaries are offering floral tribute to the idol of Padma Bhushan Dr. Karmavir Bhavna Patil. I also request all the dignitaries to offer floral tribute to the idol of Sir Lakshmidhar Bhavna who stood silent proudly and supported his noble deeds.
Please sit down. Good morning.
Good morning, sir. मी मान्यवरांना विनंती करते की त्यांनी व्यासपीठावर स्थानापन्न व्हावं. I request all the participants to maintain silence. A very good morning to all the dignitaries, guests, and delegates. With great joy and immense exultation, it gives me an immense pleasure to extend a very warm welcome on behalf of the Rayat Shikshan Sanstha's Anna Saheb Aute, Arts, Commerce, and Hutatma Babagino Science College, Manchar, who has clubbed all of us for inaugural function on the occasion of one-day international conference on exploring innovation in life sciences. Today, we have with us guests from education field to reflect upon the theme of our conference. We have Honorable Keynote Speaker, Professor Dr. S.B. Patil Sir, Principal, Dadasaheb Jyotiram, Godse Arts, Commerce, Science College, Vadud, Satara. Honorable President of this function, Dr. N.S. Gaikwad, Principal, Anna Saheb Aute College, Manchar. We also have with us Professor Dr. P.M. Dikhe, Principal, PVP College of PVP College and Senate Member, SPPU Pune. Dr. Mahesh Khade, BOS Chairman in Botany, SPPU Pune. Dr. B.D. Ranpise, Vice Principal, PVP College. Dr. Vinayak Lokhande, BOS Member in Botany, SPPU Pune. Professor Dr. K.N. Kumar, Retired Professor, SPPU Pune. On behalf of Anna Saheb Aute College, I once again extend a very warm welcome to you all, sir. I also welcome all the teachers and students delegates, research scholars, young scientists from India and abroad present here and online. I also welcome all the students once again. Moving ahead, I would like to request Professor Mr. V.S. Kumawat, Organizing Secretary of this conference, to come forward and set a preamble for this conference as well and to introduce the guest.
Hello, good morning to you all. Myself, Vilas Kumawat, Associate Professor in Botany. Rayat Shikshan Sanstha, Anna Saheb Aute College, Manchar, Department of Botany and Department of Zoology organized today one-day international conference on exploring innovations in life sciences. I warmly welcome to our principal Dr. N.S. Gaikwad sir, today's function as a president. Also, I heartily welcome to our chief guest and keynote address speaker, Principal S.B. Patil sir, my teacher, Professor Dr. K.N. Rumal sir, our today's guest, Principal Dr. Pradeep Dige sir, Board of Studies member, Kherde sir, then and his colleagues, our Vice Principal, Professor S.N. Bolbot sir, Head of the Department, Professor Pokre S.T. sir, all the heads of the department, and my colleagues, various participants from the our various states of India, resource persons, the researchers, participants, and dear students. I will briefly introduce our mother organization, that is Red Shikshan Samstha. You know as well, this is very famous for the in Maharashtra in the education. That is founder of Red Shikshan Samstha is Dr. Kamil Bhaurav Patil. Samstha established in 1919. At present, today there are 675 branches which are working in Maharashtra as well as in adjoining states of Karnataka. There are 41 senior colleges and it is recognized as the biggest education institute in Asia. Total number of employees working in our mother institution, that is 16,172. The total students studying in various branches of Raj Shikshan Sanstha, which includes high school, junior college, senior college, one engineering college, B.Ed colleges, ashram shalas, etc. That is 4,42,294 students. That is the biggest students involved in the education. Recently, Karmir Bhaurav Patil University, Satara, is the cluster university established by the government of Maharashtra on 27th October 2021 as the recommendation of RUSA, MHRD New Delhi. And we have the proud, our Raj Shikshan Sanstha now become a university, that is Karmir Bhaurav Patil Cluster University. That is our biggest achievement in our mother institute. I will briefly tell you about the Anna Sahib College, Mansar. This college established in 1966. The total student strength in this academic year is 2,121. We have UG courses run, there are 17 UG courses, and vocational courses are two, that is food processing and quality management, account and taxation. Now these courses are very good courses, that is students are benefited from this area. PG courses in our college run by total numbers are 9. You know, recently we have faced the reaccreditation for NAC. This college is reaccredited by NAC with A grade, and we have maintained, sustained the A grade. Department of Botany have PG and Research Center. Department of Botany and Zoology have published a book as a flora and fauna of Anna Sahib College campus, and this is good book. Last in 2022 we have published this book because college have 105 acres land and which is conserved for the social forestry. That is our biggest highlights of our college campus, and also the horticulture. Recently, in this academic year, college have taken initiative in collaboration with the Mansar Nagar Parishad, that is Miyawaki Udyan, which is of 6-acre land, where we plan plantation of indigenous plants. And uh, our seniors are guiding us for the development of this Miyawaki Udyan. About explaining today's in exploring innovations in life sciences, for this international conference, we got huge response from participants, sir. There are 325 participants registered for this one-day international conference. I am proud of this huge response from the various subjects from India. There are 140 abstracts we just received today. We are going to publish these research papers in UGC Careless Second, Web Science, Scopus listed journals. And this is very good achievements of the today's conference. You know, in the 21st century, life sciences become very immense importance for the mankind. We all and world just suffer from the COVID-19 pandemic, but knowledge of life sciences have helped us a lot because during this we have learned importance of medicinal plants, oxygen, that is only the main function of plants is released. Knowledge of microbiology, we learn its application. Various techniques, test COVID-19 test. That is the technique we learn from biotechnology, molecular biology, importance of medicinal plants. The giloid, that is, Spora cordifolia plant, that having immense importance. Number of people rushing for the medicinal plants to protect from the COVID-19, to develop immunity, etc. And life science nowadays in 21st century become very important, as well as technology in life sciences. Life sciences, this is the new emerging branch in today's situation. And number of students also, they are interested in life science, even though there is no discrepancy, botany, zoology, etc. Now, national education policy, that is a multidisciplinary approach. We have kept for students. Students have the opportunities to select his subject interest. And that's why we have prepared the national education policy foundation for the PG section. And in coming academic year, we are also facing the new education policy achievements. I hope this conference will helpful for various researchers, students, stakeholders. And this is my short preamble for today's seminar. All are welcome by heart cordially. Students, chief guests, participants from the main speakers from the foreign country, they are online delivering their lectures. I heartily welcome in the college campus of Anna Sebauti College. Thank you very much.
To make this occasion more auspicious, I would like to request our Honorable Principal, Dr. N.S. Gaikwad, sir, to welcome our keynote speaker, Honorable Dr. S.B. Patil, sir, by presenting a floral bouquet as a symbol of our gratitude and respect. Please have a big hand of applause. I now request Honorable Principal to welcome Professor Dr. P.M. Tikhe, Principal, PVP College and Senate Member, SPPO Pune. Request our Principal to welcome Dr. Mahesh Kherde, sir, BOS Chairman in Botany, SPPO Pune. To also welcome Professor Dr. K. N. Kumar, retired Principal, Savitribai Phule, Pune University, Pune. I request convener of this program, Dr. S. N. Bolkar, to welcome Dr. V. D. Tansise, Vice Principal, PVP College. Dr. Bolkar, to also welcome Dr. Vinayak Lokande, US member in Botany, SPPU Pune. I request Dr. S. N. Bolkar, sir, to welcome Honorable Principal Dr. N. S. Gaikwad, sir, who is our support system. I request Dr. Bolkar, sir, to welcome Honorable Principal Dr. N. S. Gaikwad, sir. Thank you all. As we are very much eager to sink into the depth of knowledge, I consider it a great honor to welcome Dr. S.B. Patil, sir, to guide us and officially mark the commencement of this conference. Over to you, sir. Dr. S.B. Patil, sir.
projector down. Pen drive. Enter.
I request all the participants to maintain silence.
Dignitaries on the dais. First, thanks to Honorable Principal, Anna Sahib College, Honorable Principal A.N.S. Gaikwad sir, invite me to deliver a keynote address. On the dais with us, Honorable K.N. Dhumar sir, ex-head of the Department of Botany, Pune University, Pune. Honorable Principal Dege sir, Honorable Khade, Chairman BOS Botany, Pune University, Pune. The convener of the today's seminar, the conference, Honorable Olbert sir, Organizing Secretary, Honorable Pokre sir, all staff members of the Anna Sahib Avte College, Mansar, delegates from all over India, and dear students. I have a great honor to say the topic which was chosen for today's conference is very excellent, have a broad exposure. That is, the exploring innovations in life sciences. So you know very well. Next one. All of you know very well that the science has always been built a movement or the built on the innovations because of it's all about the discovery. If our conceptions of biology, physics, chemistry, and other subjects in the field of science have not changed since they were first developed, we would not have made any new discovery, and our understanding of the peoples and the world not have advanced. Next one. As concerned with the benefits of the science, particularly the innovations in science, life sciences, is drastically changing day by day. However, we study the diagnose and treat health conditions. As a life science and technology rapidly advance, many researchers and healthcare provides a leveling the latest big thoughts for the benefits of their patients. That's particularly related to the healthcare. Here are some of the key benefits of life as a concern with the innovations in the life sciences. Next one. The benefits of life science innovations is a drastic. Next one. Now, this innovation helps us for the life sciences innovations in the medical, as a concern with the medical practitioners, are now able to diagnose illness with great accuracy and precision. For example, the genetic testing and imaging testings are allowing the doctors to identify the diseases and more accurately, leading to better treatment. Next one. With the help of life science innovations, the medical practitioners have now accessed the treatment that are more effective before and for personalized medicines to advanced surgical techniques. The life science innovations provide doctors with the tools they need to treat the patients with a great efficacy. Yes. For the better quality of life, as we concern with the life science innovations, has enabled to development of treatments and therapies that improve the quality of life for those suffering from the wide range of illness. For example, to all of you know about the stem cell therapy, and the research is going on that the stem cell therapy to grow some organs, as well as you know that the research regarding the stem cell technology, it is possible to treat certain types of diseases. Now, the particular, if you take the example, the cancer disease, nowadays the cancer disease, you know very well that. So, for the certain types of cancer diseases, the stem cell technology is very important. It plays vital role to cover these diseases more effectively. Another one, that is the 3D printing technology, has also opened the door-to-door personalized, the particularly to the donors or the particular organ donors and the orthopedics. It means the orthopedics particular. Next one. Now it reduced, because of this, the life science innovations has made healthcare more affordable for the patients by using the technology to the streamline process and reduce the cost. Healthcare providers are able to pass saving on their patients. Next. If we concern with the agriculture, now all of you know very well that nowadays the agriculture technology or the innovations in life sciences concerned with agriculture, we available in different forms. That is, the innovations has improved the crop yield. Now everybody can think about that, how the crop yield is growing day by day, and it become a nutritive, and also it helps to cure the disease also, or to increase the resistance of the body. That everybody can think about that, and that is a particular innovation in the as concerned with the life science innovation. And you know that the crop, everybody thinking about the to yield the crop increase more day by day to fill the food gap. So the crop yield, and it increase the food safety. Now that food which is produced through the innovations, that can increase the safety. It will become a safety, as well as enabling the development of new breeds in case of animals. That also, for the products are available from the animals, that these products are can be grown day by day. So the new breeds and the animals and plants, it has enabled the life use genetic engineering to create more nutritive food, actually, that are the nutrition of food, as well as the use of biotechnology to combat the crop diseases. Next one. Life science innovations has enabled the development of new products. Everybody can think about that day by day to how to increase the products, such as the biodegradable plastics and energy efficient lighting. It has also enabled the development of manufacturing methods and materials, the 3D printing technology, which is revolving the products and designing and it produce. Let's go. The new innovations as concerned, that the expense, these are actually it is an expensive field, and new innovations are constantly emerging. Here's the look at some of the most exciting development of life sciences that are resolving the way understanding and interact with the world around us. The researchers are now able to grow full functional organs. That is the stem cell technology I already informed you, in the lab using a patient's own cell. Now this is another one that the scientists or the researchers can work how to grow the organs. For example, you know that nowadays it is possible the liver can grow, pancreas can grow, and the other organs are they thinking about that in the laboratories by using the own cells of that particular patient or the person or the individual, and the organ can be extended. That is particularly, that is as like a regeneration, you remember that. In case of number of examples, you know very well that how the regeneration is occurs. So the nowadays, if that nowadays that how the entire organs can be grown. In case of lizard, you know that, or the crab, there is a regeneration of organs. Like that, the scientists or the researchers have worked on how to regenerate or regrowth of the organs. So by using this in the laboratories, the techniques that is, but actually it is a life science innovation to grow the organs. But nowadays, is by using the particular that own patients, the cells can be used to regrow the organs. This could be make organ transplant, and it is nowadays the transplantation of organs. You remember that. That is also, you know very well that in the accidental cases, sometimes you learn in the newspapers that the donation of organs nowadays is spreading. How to donate the organs, and that organs can be transferred into the other person, maybe young persons or their patients. That technology is actually, it is not in the only in the field of science, but it is actually the new innovation of life sciences. This could be make organ transplantation. It is nowadays it is easy or the easier and more accessible, as well as potentially save the lives of constantly people waiting for an organ donation. And nowadays the government has started the movement to donate the organs, and these organs are useful to the young generation. For the young generation, you remember, you know that, and that's why the donation organ, that movement of organ donation is nowadays going on. Next one. Now the 3D printing technology. The 3D printing technology already been used to create a variety of medical devices, ranging the, it means in case of prosthetic organs, to transport the organs or to implant the organs. For that purpose, this 3D printing technology is used, and this technology has potential to revolutionize or the medical device to create to prepare the medical devices, create and use while making them affordable and accessible. How it is the 3D technology is easily used in the field of medical science, you know. Next one. Next one. The synthetic biology is the field of study that involves creating artificial living organisms for stage. This could include creating bacteria. You know the bacteria nowadays, number of bacteria are used in different fields to grow and to create that bacteria could produce the medicines. Some bacteria are used to produce the medicines or the fuels or creating new organisms. That is, some bacteria are used for that. Also, you know very well that the bacteria are also used to kill the pesticides, or to cure the diseases, as become the medicinal bacteria like that. They may be used. While still it is early stage, in the early, this technology has potential to revolutionize the way and approach the diseases and environmental issues. Solve the environmental issues. For that, the bacteria are used. Next one. Next one. The artificial intelligence. Now everybody can see about the artificial intelligence. That is also the new innovation in life sciences. So that artificial intelligence has the potential to revolutionize the way we diagnose, treat. The algorithms are being used to quickly process vast amount of data to detect the patterns and diagnose the diseases. For that also the artificial intelligence is used the faster than before. It could also be used to create personalized treatments for individual patients and allowing more accurate and effective treatments. Next one. In the industry or in the business also that plays an important role to driving the life science innovations by providing valuable resources such a capital or the such as the capital, access the technology and expertise business can help the life science. The innovators are develop and commercialize their products. For that also the life science innovations can help. Next one. The growth of life science innovations has been driven by the increased affordable technology and rise the marker movements, which we call as that, with the advanced printing technology, the low cost and open source softwares, it has become much easier and more affordable for the development to create and develop the new products and the technologies. Next one. The future of life science innovations, I think about that, is broad, bright, and full of potential. The advance in biotechnology, nanotechnology, and artificial intelligence are creating a new world of possible for the life sciences. With the help of those powerful tools, the scientists can create new treatment and therapies for the wide range of diseases, and the conditions lead to better health outcomes for the patients. With gene editing, the scientists can create new treatment and therapies that allow tolerated to the individual patients, leading to better outcomes. In addition, the gene editing can help use to create genetically modified crops and animals that are more resistant to diseases and pests, leading to greater yield and lower cost for the farmers. That is actually the outcome of life science innovations. Life science innovation has had the profound impact on the way we live and interact with the world around us. For from advances in medical treatments, diagnostics, and advancement in the agriculture technology, life science innovations are revolutionized the way approach and interact with the environment and the world around us. So the life science innovations, that how it is in the science, how it is useful, new innovations may be used in the field of medical, agriculture, and entire target is that to how to increase the productivity, which is resistant, and provide the facilities to the peoples. So that is actually the aim of this science innovations. Nowadays, it is a broad subject, and that subject was choose by the organizers for today's conference. It is very important one, and it is actually useful to all the peoples, particularly to the budding scientists, researchers, and students in the field of science. With this, I conclude my speech, and I again thanks to the organizers and our the principal of Anna Sahib Opti College, Mansar, for giving me an opportunity. Thank you.
Thank you very much, sir, for your guidance. Moving ahead.
With this, I declare the conference is open, inaugurated on behalf of Anna Sahib Opti College, Mansar, in presence of Honorable Principal, Honorable Dumar sir, Honorable Convener, and all staff members and participants. Thank you.
Thank you very much, sir, for commencing the conference. Now, moving ahead, I would request all the dignitaries on the dais to publish a book on a reference entitled A Reference Book on Life Sciences for UGC, CSIR, NET and SET exam. Editor, Professor Dr. K.N. Dumar, and Chief Editor, Dr. Syed Ilyas. Please give a big round of applause. Honorable dignitaries on the dais to publish the book. The book is entitled A Reference Book on Life Sciences for UGC, CSIR, NET and SET examination. Editor, Professor Dr. K.N. Dumar, Chief Editor, Dr. Syed Ilyas. Now, I also take this opportunity to request the dignitaries on the dais to publish our conference abstract book with ISBN number. Please have a big round of applause. Thank you, all the dignitaries on the dais. I now welcome Honorable Principal Dr. N.S. Gaikwad sir, President of the inaugural ceremony, to give us the presidential address. I request Dr. K.N. Dhumar sir to share his words of wisdom on this occasion of international conference with us. Dr. K.N. Dhumar sir.
Very good morning to all the participants, dear dignitaries on the dais, that I congratulate all the organizers and principal of this college for organizing this giant conference as an international conference on life sciences. Generally, it is said that, or it is a fact, life science, the first 21st century belongs to life sciences. हे शतक संपूर्ण लाईफ सायन्स आहे आणि म्हणून या थीमवर आपण जे कॉन्फरन्स केले याच्याबद्दल शतशः आपल्या सगळ्या टीमला धन्यवाद आहेत. कारण हे फार अवघड आहे. पुण्यामधल्या फर्ग्युसन SP कॉलेजला हे शक्य झालं नाही. ते गायकवाड सरांनी मंचरला केलं आणि म्हणून त्यांचं अभिनंदन. मला कल्पना आहे खेडेगावात मुलं कशी आहेत, स्टाफ कसा आहे, हे सगळ्या अडचणीचाच डोंगर. सोयीस्कर अशी एकही गोष्ट नाही आणि त्या सगळ्या दऱ्या खोऱ्या पार करून मग आपण यशाकडे जात असतात. तुम्हाला खूप खूप धन्यवाद. मला बोलवल्याबद्दल सगळ्यांचे आभार मानतो. विशेषतः बोलबट सर हे माझे BSc चे विद्यार्थी, MSc चे विद्यार्थी आणि PhD चे पण विद्यार्थी, अशा प्रकारचे विद्यार्थी घडवणं हे आयुष्यामध्ये मी काम केलं. आतापर्यंत जवळ 50 विद्यार्थ्यांना PhD माझ्या अंडर मिळालेली आहे मी आयुष्यात. आणि हे जे पुस्तक आहे हे 201वं पुस्तक आहे. आतापर्यंत 200 झाले आणि हे 201वं पुस्तक आपल्या सगळ्याच्या साक्षीने इथं प्रकाशित होत आहे. याचा मला खूप खूप अभिमान वाटतो. या पुस्तकामध्ये CSR नेट आणि सेटचा संपूर्ण सिलेबस एका ठिकाणी आहे, लाईफ सायन्स. मी आग्रह धरत होतो की अशा प्रकारचं पुस्तक ह्या असल्या युनिक कॉन्फरन्स मध्ये पब्लिश व्हावं अशी माझी इच्छा होती. आणि मग मी सरांना नेहमी सांगत होतो इथं मला करायचंय, आपण काय परवानगी देणार का? सर म्हणले घरच्या माणसाला कशाला परवानगी लागते, तुम्ही या आणि हक्काने पब्लिश करा. हा भाग महत्वाचा आहे. आम्ही अनेक ठिकाणी जातो पण आपल्या माणसात आलेला हा वेगळा आनंद असतो आणि इथं ह्या कॉलेजमध्ये आल्यानंतर आपल्या घरी आल्यासारखं वाटतं, ते कुठेही गेल्यानंतर वाटत नाही. हा फार महत्वाचा भाग आहे. या पुस्तकामध्ये 13 युनिट आहेत आणि 96 चॅप्टर आहेत, 96 चॅप्टर आहेत की जे पुरे नेट सेटला आहेत. अनेक दिवसाची विद्यार्थ्यांची खंत होती, सर पुस्तकं मिळत नाही, मटेरियल नाही, लिटरेचर नाही. कारण याच्यामध्ये बॉटनी आहे, झुलॉजी आहे, मायक्रोबायोलॉजी आणि बायोटेक्नॉलॉजी, 4 विषय, 96 चॅप्टर, 52 ऑथर. ऑल ओव्हर इंडियातले 50 सिलेक्टेड ऑथर आहेत. त्यांनी हे कंपाईल केलं आणि आम्ही एडिटिंग केलेले. 2 वर्ष हे काम चालत होतं. मला खूप खूप अभिमान वाटतो की आपल्या समोर, मला ही इच्छा होती की माझ्या लास्ट खेड्यातल्या मुलासमोर हे पुस्तक प्रकाशित व्हावं, विमोचन व्हावं आणि ती इच्छा पूर्ण झाली आणि ती प्राचार्यांनी केली. ह्याबद्दल धन्यवाद. थँक्यू व्हेरी मच.
The words of appreciation and giving us the opportunity to publish the book. I now request Honorable Principal Dr. N.S. Gaikwad sir, President of the inaugural ceremony, to have presidential address and guide us with his wisdom. Dr. N.S. Gaikwad.
Good morning, everybody. Honorable dignitaries on the dais, who left the dais actually because of their hectic schedule, and those dignitaries were Principal PM Digesar, who is also Senate member, Dr. Kharde, Dr. Ranpise, Board of Studies member, actual present dignitaries, in which Honorable Professor K.N. Dhumar, who is guide of so many students who are working as faculty, including Dr. Bolbhat, and so many colleagues. they are working elsewhere also. Dr. S.B. Patil, who is also alumnus of Rayat Shikshan Sanstha. His elder brother was colleague at this college also. He is well-known writer in English, and these people, they are somehow connected with Rayat Shikshan Sanstha. Dr. Lokhande, who is now DF member of Botany, all my local colleagues, Dr. Bolbert, who is head of the Department of Botany, Professor Pokle, Department of Zoology, HOD, Professor Kumawat, all my colleagues from life sciences, and my dear student friends. This is very nice occasion to interact with the international dignitaries on some kind of things and themes, just like exploring innovations in life sciences. The topic is very vast, and I want to put up the emphasis on some of the topics related with the life sciences so that our students, our staff members, our teachers, our professors, they could grasp the things, they could digest all those things, and they could work for their betterment in life sciences as well as to do better career in life sciences. If we look at the present scenario in which the keynote speaker, Dr. S.B. Patil, he has lightened some of the things related with how the healthcare industry, how there will be some kind of artificial things will change the life of the human being, how there will be various types of innovations in biological and life sciences. If we look at the total industry, in my opinion, as being physicist and related with some kind of computer industry, so I think that global artificial intelligence, it will be playing very vital role in these kind of days where the market of this healthcare industry, it will be almost 164 billion dollars, American dollars. So there is vast scope to work in artificial intelligence and artificial intelligence related with the healthcare. If we look at the medical field side, if we look at the pharmaceutical industries, if we look at these pharmaceutical as well as medical technologies, so there will be a lot of scope for these industries, and they will be finding the right manpower for these kind of things. We generally think that only the people from computer science, or if we look at those people who are working in computer industry or software industry, they will be only as the graduate from this engineering background, and they will be having, suppose, the software type of development things. But in my opinion, artificial intelligence, it has opened the field field to all the sectors, and especially if we are the students from life sciences, if we are the students from, say, pharmaceutical industry, if we are the students from, say, medical type of healthcare type of sector, if we are the students from botany, zoology, biotechnology, so there is vast opportunities there in artificial intelligence technologies. And we know that the drug delivery sector is playing very nice role in order to maintain the patients and making the comfort to those patients. But nowadays, artificial intelligence, it is playing very nice role in drug delivery. So presently, doctors, they are providing the prescription, and they used to provide the drugs as per their opinion. But if we study these kind of things very precisely, so there will be no need of doctors to have the variability of all those things. If the patient is known, if the disease is known, if the drugs are known, so what kind of delivery should be given to a particular patient? There will be no excessive drugs to the patient, and there will be no harm to the patient on those kind of things. Uh, Another field is also emerging, just like virtual healthcare. So this virtual healthcare, that means we are all thinking about nowadays we are using this social media and all those things. Okay, so this is some kind of virtual type of thing. In healthcare also, if we need some help of say American doctors, American pharmaceuticals, and those kind of things, so this virtuality will also play to make proper decisions on these patients treatment, as well as for some kind of artificial intelligence type of technological surgeries of the uh, patients. Nowadays, big data, it is the key word, and big data in life sciences is a very vast field. Now, if we consider the big data, so big data, that means, for example, if we are having some kind of things related with our flora and fauna, okay, some kind of things related with pharmaceutical, some kind of drugs, all kind of things, they are to be accumulated together, and that data is to be managed. If we are able to manage all this data, if we are able to manage all these things, so that data, it will be very useful for taking proper decisions. And decisions in healthcare, decisions in forest conservation, decisions in making the proper, for example, plants or trees which will be supporting for the Ayurvedic type of drugs. So all these things will be managed with the big data. So big data field, it is very important in taking the decisions which will be useful on available data. And that's why this industry is having very nice scope in earlier few years. Then another field related with this big data, it is the real world data. For example, if we are having the treatment under some of the good doctors, and the patient's data is to be maintained. For example, a particular disease, particular patient, and the total data is available with some kind of industry. So they will be making proper decisions related with the drugs. There will be very good type of decisions in healthcare, and this will be very useful for the proper drug development also. So these kind of things are uh, available as the part of research or innovations or the recent trends in the life sciences. I will again come to a last point, and that will be related with sustainable development goals, which are prescribed by the United Nations. And you know that there are certain goals related with the protection of the earth. The one of the goals, goal 14, is related with life under water. So we know that two-thirds of the earth is with the water, and life underwater is very important. So that life should be protected properly, and these kind of artificial intelligence type of big data analysis, it will be also supportive to this sustainable development goal life underwater, which will be a 14th number. The 15th number sustainable development goal, it is to protect, restore and promote use of terrestrial ecosystem, that means life on the soil. So life on the soil, you know that the forests are there, plants are there, medical, all those things are there. Any kind of uh, fertilizers, nowadays we know that some kind of green fertilizers are used. So you will have to manage all those things, and you will have to throw some kind of poisonous things away, so that the next generations, they will be having good health in their generations which are coming ahead. So all these things, they are related with the life sciences, and this is the conference, international conference, in which we will have to learn about all those things. So this is very nice opportunity that Professor K.N. Dhumal sir, he has given us the opportunity to publish his reference book, which is related with life sciences, and it will be useful for the aspirants who are facing the NET or SET, NET and SET type of examinations, or maybe some kind of another competitive examinations. So I will be very much thankful to Dr. Dhumal, and my another friend, colleague, Dr. S.B. Patil. He has spared his valuable time for providing the keynote address, and he has also given emphasis on the healthcare sector and what could be the new trends in healthcare sector. So I again must thank you, my colleagues, for organizing this conference, one-day conference, and I shall be very thankful after some of the time, some of the colleagues who are international speakers, they will also talk virtually with you people, and you will have the facility to interact with those kind of eminent scientists in life sciences. Thank you very much for all these things, and I wish we will take some good things from this conference, and my new aspirant students, they will like this conference very much. Thank you very much.
The presidential speech. Before moving ahead to the plenary sessions, next sessions, I take this opportunity to request Honorable Principal on the dais to welcome the judges for oral and poster presentations. I would like to request Honorable Principal Sir to welcome Dr. Jamadar Sir, ACS College, Narayana, who will be judging the oral and poster presentations for today's conference. Dr. K.N. Dhumal Sir is presenting a floral bouquet. Dr. I would also request Professor Dr. Dhumal Sir to welcome next judge, Dr. Nitnaure, Madam, from HRM College, K, Rajgurunagar. Welcome, ma'am. Thank you, sir. Thank you, ma'am. At the end, I request Ms. Hemangli Kavit ma'am to conclude today's inaugural function of the conference. Ms. Hemangli ma'am. Thank you. Thank you, ma'am. I'm very much pleased to be standing here on behalf of Annasai Rauke College, Manchar, and staff to convey my gratitude for this inaugural session. Firstly, I would like to express my appreciation to our keynote speaker for sparing his valuable time to address the gathering and for highlighting the issues that we need to deliberate on during the conference. Next, I would like to thank Professor Dr. K.N. Dhumar sir for giving us the opportunity to publish the book and sharing his insights and sharing the words of appreciation as well. I would like to thank Mr. V.S. Kumawat sir, organizing secretary for this conference, for setting the preface and introducing the guest. I would like to thank the dignitaries present over here for inaugural session, Professor Dr. P.M. Dighe, Dr. Mahesh Khade sir, Dr. B.D. Rantise sir, Dr. Vinayak Lokhande sir, for sparing their valuable time and gracing this event with their presence. Next, I would like to thank the entire organizing team, Dr. S.N. Bolbert sir, convener for today's conference, Mr. S.T. Bokle sir, coordinator for this conference, all the organizing staff, members, and non-teaching staff, support staff. Last but not the least, I would like to thank Honorable Principal Dr. N.S. Gaikwad sir for his kind guidance for making this conference happen in our college and also in inspiring us to host it successfully. Thank you very much, sir. And also, I would like to thank all the audience, student members, vice principal, and our fellow staff members for to be present over here for this inaugural function. Thank you very much, and we look forward to an extremely productive discussions ahead. On behalf of organizers, I conclude the inaugural session. Thank you very much.
After auspicious inaugural session, let's revert to the plenary session. For session first. Wait, hold. After auspicious inaugural session, let us revert to the plenary session. For the session, I would request to Dr. B.H. Lokhande sir to chair the session. I request to Dr. B.H. Lokhande sir to chair the session. Thank you, sir.
The guest speaker for the first session is Dr. Gurmant Patil sir. I request Dr. Lokhande sir to chair this session. Dr. Lokhande. I request the participants to please maintain silence. Over to you, sir.
Thank you. On behalf of International Conference on Exploring Innovations in Life Sciences, organized by Department of Botany and Department of Zoology of Anna Sai Aute Arts Commerce Science College, Mansar. I wish very warm welcome to my dear friend and the today's resource person, Dr. Gunwant Patil. I will just briefly introduce Dr. Gunwant Patil, and after the introduction, he will have the session and his talk. Dr. Gunwant Patil currently is working as an assistant professor at IGCST, Texas Tech University, Lubbock, USA. He's working as an assistant professor at the Institute of Genomics for Crop Harvest Stress Tolerance and Department of Plant and Soil Sciences at Texas Tech University. Dr. Patil, he has received his BSc degree from North Maharashtra University, Jalgaon, and his MS and PhD degree he has completed from the University of Pune and National Center for Plant Biotechnology, New Delhi, India. Prior to joining to the Tech faculty, Dr. Patil, he has served as a research scientist at the University of Minnesota, Paul. He also worked as a postdoctoral associate at the University of Missouri, Columbia, and a visiting researcher at the Swedish University of Agricultural Sciences in Uppsala, Sweden. And on his credit, he has published more than 50 research papers in highly reputed international journals like the Plant Science, then the Scientific Reports, the Physiology, etc. With this brief introduction, I wish very warm welcome and would like to invite Dr. Gunwant Patil to deliver this talk.
Right. Thanks, Vinayak. Yeah.
Is it audible to you, Dr. Gunwant, sir?
Yes, I can hear you. Can you hear me?
Yes, yes, yes.
All right. So I will share my slides, and can you see my slides?
Yes. Volume.
All right. So I will just go ahead and start. Let me put it here. All right. So thank you, Vinayak, for this great opportunity, and also thanks to Sachin Pawar. So both Vinayak and Sachin are my good friends, and thanks for invitation. And I'm also glad to see Dhuma sir on the stage. He is my like a
Dr.
teacher.
Gunwant, sir.
Sorry.
Can you raise your volume on your side?
All right, let me see. Okay, can you hear me now?
Yes, yes.
All right. So again, like thank you, to Vinayak and Pawar for this invitation to deliver talk to your conference, and also I'm glad to see Dhuma sir on the stage as he was my teacher during my masters. So again, my name is Gunwant Patil, and I'm assistant professor here at Texas Tech University. And today I'm going to talk about the research that we are doing for last 4 to 5 years on developing a sustainable technologies to improve nutrient uptake in crops through single cell genomics and gene editing tools. So before I start, I would like to give a very brief introduction to my lab. So this is my lab, and I have like a couple of PhD students working with me on different topics of genome engineering, genomics, and phenomics. And I also have like several postdocs working on the different area of crop improvement. And here we are located here at West Texas in a city of Lubbock. And this is our institute, which is known as the Institute of Genomics for Crop Abiotic Stress Tolerance. So my lab at the Texas Tech University work on the two major areas. The first area that we work on trait discovery, where we are integrating the genomics and genome engineering technologies to discover new traits, new technologies for improving the water stress tolerance, nutrient uptake, improving the root system architecture, understanding the beneficial interaction with the microbes, and as well as disease resistance. And also, I have like a very special interest in technology development. And my lab is also investing a lot of resources to develop a new technologies for genome engineering, especially improving the plant transformation and gene editing technologies. And as you can see on my slide, my lab particularly work on the soybean. So that's my major crop that we are working on different traits. But at the same time, I work on cotton because I grew up on the cotton farm in the district of Jalgaon. So the cotton is very much close to me. And I also work on sorghum. At the same time, we also work on a different models plants, including tobacco, Arabidopsis, and Ceteria. So these are the model plants which are very easy to transform and do the basic gene discovery and trait improvement. But for today's talk, I'm going to mainly focus on how we how we are using the single cell genomics and gene editing technology to improve the nutrient uptake in plants. And the particular focus is the soybean and sorghum. So before that, I would like to give like some background about nutrient uptake. So as we all know, nutrients are super important for plant growth and development. And we know the farmers apply a lot of nutrients to their field to improve the productivity and the seed yield. But whenever we talk about the nutrients, we mainly talk about the three nutrients like the nitrogen, phosphorus, and potassium. And of course, like these are the most important nutrients for plant growth and development. But at the same time, the mineral nutrients which are present in the soil and in the very stress amount, they also play a very important role in plant growth and development. However, these mineral nutrients are not extensively studied in crops. And we also know if there is a deficiency in a particular nutrient that can reduce the yield and growth of the plant. And also if you have the excess quantity of that nutrient, that can lead a toxicity effect on the plants. And that can also reduce the yield and growth. So therefore, we have to have a very optimum concentration of these nutrients or mineral nutrients, not only to improve the biomass and yield, but they also play a very important role in abiotic and biotic stress tolerance. And these are like some of the responses. So for example, these are the real response of the deficiency response of manganese, copper, or zinc on the soybean plant. So if these nutrient elements are in the less quantity, the plants become the chlorotic and it reduces the yield. But at the same time, if these elements are present in the high concentration, that can also lead to toxic effect on the plant growth and development. And so why we should care about the mineral nutrients and to profile the mineral nutrients in the germplasm. And this is the reason. For example, if you have like a lot of varieties of a particular crop, and if you know some of the varieties has a low accumulation of particular mineral nutrient versus some of the lines which has the high mineral nutrient content. So based on the soil condition, so if your soil condition has a deficiency, you can use the lines which are high accumulator of particular nutrient. And if you have the toxicity, you can use those lines which has the reduced level of mineral nutrients. So this is very important for us to identify the nutrient uptake in the germplasm that we are growing in our field. So this nutrient uptake is very essential. And for example, if you have the uniform soil nutrient content in your field, and if you're growing maybe like 10 different varieties of a soybean, and you can see like some varieties can have a better nutrient uptake efficiency, while some varieties will have less nutrient uptake efficiency. And this nutrient uptake efficiency is controlled by the genetic variation. So basically the genetic makeup of the crop or of that plant influence the nutrient uptake and the nutrient utilization efficiency. And therefore having this knowledge is very important, because if you know your plant variety has a better efficiency or low efficiency, based on that data or based on that knowledge, you can apply the fertilizer to your field. And therefore you can reduce the excess fertilizer application to the field, which is very costly, and you can it will have like a sustainable effect on your soil properties and the soil health, as well as getting more yield from the less application. And therefore, to understand the nutrient uptake efficiency and utilization efficiency, we initiated our study. In this study, we selected a diverse soybean germplasm. So here we selected about 220 diverse soybean accession, which has elite soybean lines, some land races, and wild soybean. And these lines also have a whole genome sequencing data. So we have like a very good genotypic data coming from these 220 lines. And these lines were selected from different countries all over the world. So these all 220 lines were subjected for the screening in the greenhouse condition for 2 years. And following the screening, we collected the tissue. For example, we collected the root tissue to understand the nutrient uptake. We collected the stem tissue to understand the nutrient transport. And we also collected the leaf tissue to understand the nutrient deposition. So understanding the nutrient uptake and deposition is very essential to see which variety or which line has a better capacity or the genetic mechanism to uptake the nutrients. So following this tissue collection, we perform the X-ray fluorescence analysis. So this is the instrument that we use to analyze the mineral nutrients in the leaf tissue or the root tissue. So altogether, you can see like we collected about 2,600 data points from this study. So it's not only like we perform this study in the greenhouse, but we also perform this study in the field condition to see the selected lines really perform well in the field condition. So we grow these lines at South Carolina with our collaborators and also at the Texas Tech University farms. So based on this greenhouse and the field study, we identified a very significant variation for at least 10 mineral nutrients from our germplasm. And not only that, we were able to identify some of the lines which are low accumulator of different mineral nutrients like silicon, zinc, sulfur, and other things. And we also identify some lines which shows a very high accumulation of these mineral nutrients. So this data is very important for the soybean breeder and also farmers so that they can select the correct variety to grow in their field. So this study just published like a few months back in Environmental and Experimental Botany. And since we have like a really good genotypic and phenotypic data, we perform the genome-wide association mapping. So this is like basically association mapping to map the genomic loci associated with that particular trait. So using this analysis, we identify a lot of important QTLs or the genomic loci essential for uptake of the aluminum in the soybean leaf, uptake of silicon, uptake of iron, and manganese. So we identify a lot of different QTLs and underlying genes for these nutrients, mineral nutrients from our study. And if you are interested, you can read more about this study. So since we have like a lot of mineral nutrients identified in our study, I'm going to focus mainly on the silicon and how we characterize this particular trait in more detail and identify the genes and transporter responsible for silicon uptake in soybean. So why we are interested in silicon? And the thing is like silicon is considered as a beneficial nutrient. And as we all know, silicon is the second most abundant element on the earth crust. After oxygen, silicon is the most abundant element on the earth. And this silicon is uptaken by the plants in the form of silicic or monosilicic acid. So this is the soluble form of silicon which is uptaken by the plant. But it's not like all the plants can uptake the silicon. The plants requires a specific mechanism, genetic mechanism, or specific transporters to uptake the silicon from their roots and transport all the way to the leaf tissue via a water stream or the water uptake. most importantly, this silicon plays a very important role in both abiotic and biotic stress. And you can see hundreds of papers that show that application of the silicon can improve the disease resistance. For example, in rice, it can play a very important role in fungal pathogen. In the same way, it can play an important role in soybean against the Phytophthora shoji, which is another fungal pathogen. And you can see like this is the silicon treated with, this is the soybean treated with silicon, and this is soybean without silicon treatment. And you can see a very beneficial effect of the silicon application on the plants. At the same time, this silicon play a very important role in abiotic stress. And some of the studies are from my lab and my colleagues that we are studying more the effect of the silicon on the abiotic stress tolerance. Although like silicon plays an important role in abiotic and biotic stress, the underlying genetic mechanism is still unknown. And that's what we are trying to understand in our research. But what is known that when the silicon is uptaken by the plants, this silicon creates a apoplastic obstruction. So basically it creates a barrier, a physical barrier which restrict the entry of fungal pathogen or bacterial pathogen in the leaf tissue. At the same time, this physical barrier reduce the water transpiration. So basically it conserve the water. And because of this barrier, you can get the dual benefit. Like you get the abiotic stress tolerance as well as you get the biotic stress tolerance. Also, the silicon accumulation in plants is very dynamic and very differential process. So some of the plants like tomato, canola are the poor accumulator of silicon. So even though you apply a lot of silicon to these plants, they cannot accumulate the silicon because they do not have a functional transporters or the functional genes to uptake the silicon. But on the other hand, rice and sunflower are the high accumulator of silicon because they have the functional genes for silicon accumulation. And this silicon accumulation in plants or the silicon transport in plants is controlled by two major genes, LSI1 and LSI2. And these genes act as a silicon influxer and silicon effluxer. So basically, these silicon molecules are influxed by the roots to the xylem tissue. And with the water stream, the other proteins, the effluxer transporters or the proteins, transport that silicon to the leaf tissue. So this both the genes plays a mutual a mutual role in a silicon transport from taking the silicon from the roots and putting them to the leaf tissue. And this silicon uptake is well characterized in rice, but its status, its transport, and its mechanism is not well studied in soybean. And that's what my lab is trying to do. So to understand the silicon accumulation, we are growing our our soybean plants in the hydroponics condition, where we are growing the plants without silicon and with silicon. And as you can see with this scanning electron microscopy, coupled with the X-ray fluorescence, this green tissue shows the accumulation of the silicon in the leaf tissue. So this is like a very enlarged image under scanning electron microscopy. But when we compare this with the sorghum or the monocots, you can see the silicon is accumulated in a very specialized cells. We also call them as a silica cells. So as you compare the soybean, which is dicot, versus sorghum, which is monocot, you can see the evolutionary, they have like a very differential mechanism for silicon uptake. In sorghum, it is accumulated in the specialized cell, but in soybean, it is dispersed throughout the leaf tissue. And the mechanism for this accumulation is still largely unknown. And you can see like more data from this one, like this is the control. You can see no silicon with the X-ray scanning electron microscopy. But as soon as you apply the silicon, you can see the silicon accumulation in the trichomes and the epidermal cells of the soybean. And finally, after a few days, the silicon is dispersed throughout the leaf tissue. And this silicon accumulation in the leaf tissue provides the physical barrier. So basically, it gives the a barrier against the abiotic and biotic stresses. So our next question was like, what is the cellular heterogeneity involved in the silicon accumulation? So what are the cells? What are the specific tissue are involved in the silicon accumulation? And to understand that one, we perform another study where we grow the plants without silicon and with silicon. And after a few days, we perform the X-ray scanning electron microscopy, and you can see the silicon accumulation here. At the same time, we perform the single nuclei isolation to understand the cellular heterogeneity. And this is a very new technology that we have implemented in my lab. So why we are working on the single cell technology? So this is like a very powerful technology which is recently discovered in different organisms. So with the single cell RNA sequencing, you can identify the gene expression at the individual cell level. So that provides a very powerful data that you can identify the expression of the genes at each and every cells. So this is the example, like for example, this is the leaf. You are interested to see the expression pattern, and using the conventional RNA sequencing, you can identify the gene expression in the whole leaf, but you don't know these genes are expressed in which cell types. But with the single cell RNA sequencing, you can identify the expression of each and every gene in every cell type. So that provides a very like a high resolution uh, transcriptomics data from any given tissue. And at the same time, this is very good technology to identify the cell types, identify the developmental trajectory, and also identify the cell-specific gene network. So this is like a single cell technology that we have applied in my lab, and my PhD student Leo is like expert in this technology. So we basically isolate the nuclei and pass through the single cell pipeline and identify the single cell data. I'm not going to discuss in more detail, but this is like a very nice technology that can identify the cell population within the organ. So using this technology, we were able to identify about 12 different cell types in soybean. So this is the first study that shows about 12 different cell types within the leaf tissue. So with that, we were able to precisely identify the epidermal cells, vascular cells, or mesophyll cells, and also the vascular cells like xylem and phloem. And these were like a very precise to identify. And at the same time, we identify the different genes which are specifically expressed in those tissues, as you can see in this example here. One of the interesting things that we discovered from this study was when we apply this single cell technology, we were able to identify a unique and a new cell types which are specifically induced in the silicon treatment. So you can see like when we apply the silicon treatment, we were able to identify specific cells in the silicon treatment, and this is absent or pretty much absent in the control without silicon. And these cells were closely related with the xylem. And this study again confirmed that the silicon is uptaken by the roots, then transported to the xylem, and then from xylem it is transported to the epidermal cells. But we don't know like whether they are differentiated from xylem or other tissue. So this is like something that we are again trying to understand. Another interesting thing, we found that the silicon is very essential. And as I mentioned previously, like silicon is important for biotic stress. And when we apply the silicon to the plants, we identify the trigger in the biotic response genes. So as soon as we apply the silicon, the genes involved in the biotic stress or the resistance or immunity were expressed at a very high concentration. And these are the example of the cells, like we identify a cluster of cells which are involved in the immune response in plants. So it provides the immunity to the plants. We identify the genes involved in the jasmonic acid signaling, which is the essential metabolite for plant growth and development and against the biotic stress. We identify the genes involved in the defense response to biotic stimuli, and also salicylic acid response. So these are the very important genes that determine plants' health. If you have these genes expressing into the plants are healthy against, or the plants are protected against the biotic stress, like a fungal disease or the bacterial diseases. So this is the first study that shows that silicon can induce the biotic stress response. And this was again confirmed with the known genes which were published earlier. Also, coming back to the QTL that we identified on 16. So, sorry, there is like some noise in background. All right. So, as I mentioned, like using the genome-wide association mapping, we identify the 3 important genes. Sorry?
Online participants are requested to mute themselves. Online participants are requested to mute themselves.
All right. Sorry about that. All right. Anyway, like, I will continue. I think we have like a limited time here. So we identify like we identify like a 3 important genes involved in the silicon transport, and these are the transporter genes. So if these genes are expressing in the soybean, we can see the silicon is uptaken by the plants, and that was also again confirmed by the single cell data. So as you can see, like we using the single cell data, we were able to identify a precise location of the silicon expression in the epidermal cell layer. So, and this was just possible using the high resolution single cell nuclear RNA sequencing. So now we have the genes in our hand. So we want to functionally characterize these genes. And therefore, to characterize these genes, we use the gene editing technology as a tool to functionally characterize the genes. So I don't think like I need to introduce what is the gene editing, but this is a very powerful technology that can be used in the plant biotechnology, improving the livestock, improving the diseases in the humans and animals. And it has a lot of application in the drugs, in the microbial research, as well as the food industry and so on. The overall goal of the gene editing technology, like a CRISPR-Cas technology, is that we using this technology, we can edit the gene, we can delete the gene or the piece of gene, we can add the new gene, or we can also silence the gene. So this is like a really good technology that can be used to manipulate the genes very precisely. And this technology we use as a tool to functionally characterize the genes involved in the silicon uptake in soybean. So we perform the gene editing in soybean, and we develop the transgenic plants or gene edited plants using the CRISPR-Cas9. And this work was led by my postdoc Vikas Devkar in my lab. And as you can see, like when we knock out the silicon gene, the silicon accumulation was reduced significantly as compared to the wild type. So as compared to the normal plant, the silicon uptake was reduced almost like 70% in the knockout plant. So this clearly suggests that this particular gene plays a very important role in silicon accumulation, and this can be confirmed using the gene editing technology. So next thing is like this gene is involved in the silicon uptake, but does the gene plays any role in water stress or not? And therefore, to understand this, the role of this gene, we perform a small study where we grow our soybean plants in a one tray and we withhold the water for 7 days. So basically, we imposed a severe drought to the plants. And as you can see that the wild type plants, which are shown in the red text, are the normal plants, which are not gene edited. But as you can see, the white text plants or the knockout plants, they show a very severe phenotype during the water stress. So this study again confirms that these genes not only play a role in silicon uptake, but they also have an important role in abiotic stress tolerance. So this is like a very important study that determines the role of the genes, and that can be used directly in the field condition. Not only that one, like a silicon is a beneficial nutrient. So under normal well water condition, the knockout lines, which you can see here, they are very happy. They are as happy as the wild type. So under well water condition, there is a no negative or positive impact of the these particular genes. But as soon as you impose the drought stress, the knockout lines perform really poor. So this again confirms that these genes are very essential for providing the drought tolerance to the plant. And therefore, having this gene in the field condition, because we know like in India or all over the world, the drought is predominant like environmental stress, and that can come anytime during the plant growth and development. And having these genes is very essential to confirm the drought tolerance. So based on this study, we confirm that silicon may be not essential because it does not play a very important role during normal condition, but this silicon and the genes plays a very important and beneficial role during the stress condition, either water stress or biotic stress in terms of the fungal or bacterial infection. So again, like this is a study that concludes the importance of silicon and importance of the genes that we have discovered in our study. And therefore, to conclude, in summary, we identify the greater phenotypic variation in the nutrient uptake in soybean. We also identify the silicon uptake in soybean is controlled by the QTL known as a high-sil QTL on chromosome 16. Using the single nuclear RNA sequencing, we were able to identify 12 different cell types in the soybean leaf. We also identified the silicon treatment induced the new cell types which were related to the xylem cell function. We also characterized the gene function using the CRISPR-Cas technology. And from this study, we identified this particular gene, which is high silicon 2C, plays a functional role in silicon efflux in soybean. And this gene is specifically expressed in the epidermal cell layers, which can be identified using the single cell technology. From this study, we confirmed that the silicon treatment primes or the boost, basically this silicon treatment boosts the plants against the biotic stress. And possibly this provides the adaptive immunity to the plants. And again, like this is like a very interesting and a large area of research, and there are like still many unanswered questions that need to be answered. And we are still working on this project to answer many important biological questions, which can have a direct implication in the farmer's field. And with that, I would like to thank many collaborators, which played a very important role for this work. Basically, this work in my lab was carried by Vikas Devkar and Leonas D'Agostino, who is a PhD student in my lab. I also collaborated with Rupesh Deshmukh from Central University of Haryana, Bob Stupar from University of Minnesota, Nadab sir from Savitribai Phule Pune University, and also the funding support from Southern Soybean Board, United Soybean Board, and Texas Tech University. And also thanks to Vinayak and Sachin for invitation and providing this opportunity to talk in front of you. So with that, I'll be happy to take any question. And thank you.
Thank you, Dr. Guru. Now the session is open for the discussion. If the audience, they have a question, they can ask.
If someone has questions, they can raise their hand from the participants who are here. And online participants, if someone has questions, they can unmute their mic or send a message in the chat box.
Hello, good evening, Dr. Patil. This is Balasaheb Sonone from University of Illinois. Uh,
Hi, Bharat.
uh, I was wondering how, like, which type of cells appeared under silicon stress in xylem? Are they playing some role in terms of water transport, or they are just uh, acting as a uh, collection for silicon?
I think this is like a really good question. So the silicon transport is a very active mechanism. So it's like as long as there is a efflux of silicon, it will keep accumulating. And during this process, we still don't know the complete answer here, but what we think is like the xylem cells were differentiated to become the silicon induced cell. So basically what happens like it's a very well studied phenomena, like when the silicon is there in the roots, it can create the capsarian strips, which is like a secondary strips involved in the accumulation of secondary metabolites. So we also think like these silicon induced cells are derived from xylem, because xylem is the like the tissue for the silicon transport, and they are differentiating to the other tissue, other cell types, which we don't know yet. But the next step, so after that one is like the silicon is then unloaded to the epidermal cell layers. So the between xylem and the epidermal cell layer, these are the new cell types that we identified, and we are still trying to understand their the mechanism or the property of these new silicon induced cells.
Yes, thank you.
Yeah.
Is there any question? Actually, Dr. Gunot, I want to add something with respect to the silicon. In India, basically, whatever the fertilizers nowadays they are spraying for the water stress management, basically the fertilizers they have the silicon. But how much it is effective for the protection, even farmers they don't know, even the biofertilizer companies they don't know. But directly they are using such kind of the chemicals for the productivity and for the plant stress management. So whether it is beneficial in the due course of time for the plant as well as for the farmers, or there are some solutions.
Yeah, I think this is like really good and very important question. And the answer for this one is a mixed answer, because if you are blindly spraying the silicon to the plants, it's not going to work. And as I mentioned, like plants need a genetic mechanism to uptake the silicon. So if you are spraying the silicon to the leaf tissue and the leaf tissue do not have the mechanism to uptake that one to create the silica gel or the physical barrier, then it's not going to work. So basically spraying is not the best option. The best option is to provide the silicon from the roots, because they have the active transporters to uptake the silicon in the form of salicylic acid, then unload to the xylem stream, and then it is accumulated to the leaf tissue. So
And
there are like a lot of studies, but it's a debatable like whether it has a direct application or not. So it need to be studied in more detail.
are they interfering with the other nutrients uptake which are essential for the?
No, no, that's good thing is like silicon is like very much inert. It does not interfere with other minerals or other nutrients. So that's good thing. Yeah.
And the accumulation in the seed parts because
Yeah,
they are
that's
directly.
yes. That's another good thing is like it does not get accumulated in the seeds of many crops, but few crops it can be accumulated.
Okay. If there is no question, then I would like to conclude the session. First of all, on behalf of Anna Saivati College Mansar, who have organized this nice conference on international conference on exploring the innovations in life sciences. For this, Dr. Gunod Patil, who has delivered the lecture on the sustainable technologies to improve nutrient uptake in crops to the single cell genome and single cell and the genome engineering. He has delivered very nice talk on this current issue, which is very important for the farmer. basically he has covered all the things with the high throughput technologies, and he has focused on the plant or the crop soybean, who has not been that particular plant not explored for this silicon uptake or the silicon uptake efficiency. In his talk, he has focused on the CRISPR-Cas technology as well as the gene knockout, where he has got good results for this silicon uptake in control condition as well as stress condition. Basically, the topic represent that the under stress condition silicon is working very efficiently, and definitely when the water consumption efficiency, when we talk about that, we should take care of that particular type of the chemicals to spray on the crops or to provide the such type of the chemicals in soil for the uptake. So with this brief conclusion, I would like to thank Dr. Gunwan Patil, who has given the time to deliver this lecture, and I'm also thankful to him because now it's almost 12:30 there, 12:30 p.m. It's in night he is giving this talk, and he has waited for a long time to deliver this talk. So on behalf of Anna Sarman of Mansar College, and on behalf of me, myself, Sachin, as well as Kirti, and our Guru, Professor Bhuman sir, I would like to thank Dr. Bhuman for his nice talk. Thank you.
Thank you.
Now there is another talk will be delivered by the Dr. Balasek Sonani. He is with us virtually. So I would like to invite Dr. Balasek Sonani. Dr. Balasek, are you there? Okay. So I will briefly introduce Dr. Balasek. Basically, Dr. Balasek is also my good friend. He's working as a postdoctoral research associate at University of Illinois, Urbana, US. Balasek Sononi is joining us from University of the Illinois. He completed an MSc in Agricultural Biotechnology from the MPKV Rahuri and Marathwada Agricultural University in 2009. He was with us at Bhabha Atomic Research Center for his PG dissertation work, and during that time we met. He worked at various research institutions for the research in India. Then he received the Australian Fellowship for his PhD in Environmental Interactions of Sequel Photosynthesis at Western Sydney University in Australia. He also received the prestigious Marie Curie Actions Postdoc Fellowship from the European Union. His work primarily focused on the interaction of the plant photosynthesis and hydraulics to climate change factors such as the heat and the drought, basically working with the aquatic stress. His work was published in highly reputed international journals in the plant sciences, and overall his research contributes to the understanding the fundamental kind of the physiological process towards the developing climate resilient crops. Today, Dr. Balasek is going to talk on the exploring and utilizing the plant ecophysiological traits in improving the crop productivity and the water use efficiency. With this brief introduction, I welcome Dr. Balasek and hand over the session to deliver his lecture. Thank you.
Thank you, Dr. Vinayak Lokhande, sir, for the warm introduction and for inviting me to share my research at the conference. I am also thankful to the conference organizers for providing me an opportunity to share my research with the next generation of researchers from my homeland. So, I am talking from University of Illinois, which is located here in the part of USA, and our campus looks like this. It's a very nice campus located in the farm of corn and soybean belt of United States. In today's talk, I'm going to give research which I conducted at the Washington State University at this left right corner of United States. So in today's talk, I will share research conducted at Washington State University with my colleague, Dr. Varsha Pathare, and my collaborator, Dr. Laura Bartley, and my mentor, Dr. Asap Cousins. So let's have a quick look at the main challenges in plant sciences. Here is a synthesis of one collaborative studies conducted by researchers from Africa, Asia, Europe, and Americas. As you can see, there are two words those are prominently appearing in this word clouds. Those are climate change and crops. This study also gives a clear indication that our challenges are global. They don't have regional boundaries, and we need to find the solutions to overcome them together. We all know that what is causing the climate change. Since the invention of this commercial steam engine around 1700s, we have been burning fossil fuel continuously. Recently, we started burning more fossil fuel and emitting more carbon dioxide in the atmosphere. This carbon dioxide molecules traps heat, which is known as the greenhouse effect. Because of the greenhouse effect, our planet is warming. We have raised the Earth temperature around 1.5 degrees Celsius since industrialization. And this warming has a huge impact on rainfall patterns. Because for each degree Celsius increase in the air temperature, the water holding capacity of air increased by 7%. In other words, our atmospheric air is becoming more dry with global warming. So this dry air pulls water from land and water resources, including lakes, rivers, our plant canopies, soil system, and all these processes influences the global water cycle and rainfall patterns. And we are witnessing these changes everywhere in the world, including in India. We are observing frequent and intense drought and frequent and intense flooding events. As a consequence, our crops are facing a hot and dry climate and not producing expected amount of food for us. For example, in this figure, a continuous line is showing the projected rice yield by 2050, and the dashed line is showing the expected rice yield by 2050. And you can see there is a gap in global food supply and demand. There will be a huge gap in global food supply and demand by 2050. So this is one of the major concerns for global food security. Crop produces foods using photosynthesis. Plant photosynthesis uses atmospheric carbon dioxide and water to convert energy from sunlight into the carbohydrate and that is one of our food. And so let's have a look how plants processes this carbon dioxide and use water during the photosynthesis. So as shown in this cartoon, there are small tiny pores on the leaves called stomata, through which carbon dioxide molecule enters and water leaves from the leaf. Once CO2 or carbon dioxide enters inside the stomata, it has to cross various barriers in order to get assimilated or fixed by enzyme Rubisco inside the chloroplast. And so the easiness or ease of this carbon dioxide movement from intercellular air surface to the site of fixation is called as mesophyll conductance. Similar to carbon dioxide, water molecule also faces various barriers as it moves from the leaf xylem to the site of evaporation and the site of evaporation outside the stomata. And the ease of this movement of water from xylem to the outside of stomata is called as leaf hydraulic conductance. Both mesophyll conductance to CO2 and leaf hydraulic conductance to water are very important trait that determines the water cost for CO2 assimilation. Consequently, these two traits have major impact on plant productivity and water use efficiency. Therefore, manipulating this trait to increase CO2 uptake relative to water loss is one of the strategies to increase productivity and water use efficiency in crops. However, the main question is what to change or how to manipulate these traits. And for that, we turn to the nature. We know that grasses grows everywhere. If you grow in the, if you walk in your field, or if you walk in the mountains or hillside, you will see grasses. About 20 to 40% of Earth's surface is covered by grasses, including crops. Our main crops like wheat, rice, corn, sugarcanes, all are from grass families. And also there are wild grasses. Importantly, grasses are adapted to harsh climate as shown in this small figure. And they can survive on very low water content. So we decided to understand how these grasses survive in dry habitats. For this, we selected grass species from diverse range of rainfall. Uh, as you can see that we have selected grasses, those are growing around 250 millimeter of rainfall to 2,500 millimeter of rainfall. So there is a huge variation in terms of rainfall for our selected grasses. And then we grow these grasses in the common greenhouse experiment. We estimated mesophyll conductance, leaf hydraulic conductance to water, leaf level water use efficiency, and measured various leaf anatomical traits. As you can see, here are some representative cross-section images. These grasses showed spectacular variation in leaf anatomy. And so here I am showing the principal component analysis, which shows the relationship between various traits and mean annual precipitations. These traits separated species along the rainfall gradient. On this right-hand panel, we can see the traits are related to low rainfall, whereas the left-hand panel, we can see the traits related to high rainfall. And just to note that the traits which are located in opposite direction to each other, they show negative relationship, whereas the traits those are located close to each other or in the same panel, they have positive relationship with each other. So the key finding from this research is that mesophyll conductance to CO2 and leaf hydraulic conductance to water are negatively correlated. Now, as I mentioned earlier, mesophyll conductance determines CO2 uptake and leaf hydraulic conductance determines water use. Hence, species adapted to drier habitats also have greater water use efficiency. So this study suggested that CO2 and water movement are coordinated in the grasses. This study also identified few anatomical traits. Those can be used to manipulate the leaf hydraulic conductance and mesophyll conductance of CO2. For example, here is the one trait which is cell wall thickness. Specifically, this is the mesophyll cell wall thickness. If we have high mesophyll cell wall thickness, that will give high leaf hydraulic conductance, low mesophyll conductance to CO2, and consequently we will have low water use efficiency. We decided to use this target this trait and test, do we really get high water use efficiency if we have thin or low mesophyll cell wall thickness. For this, we have a collaborator who developed the cell wall transgenic line with thin cell wall in rice. So this is the rice transgenic lines where we have around 12% reduction in mesophyll cell wall thickness. So the transgenics has a thin cell wall. We grown this plant in greenhouse experiment and measured various physiological traits, including mesophyll conductance to CO2, net carbon assimilation, leaf hydraulic conductance to water, plant biomass, and whole plant water use efficiency. So we found that transgenic lines, which have thin mesophyll cell wall, showed increase in mesophyll conductance to CO2. Because of this increase in mesophyll conductance to CO2, we also observe increase in net CO2 assimilation rate. However, we do not see or we do not observe any changes in leaf hydraulic conductance. At whole plant scale, we observe around 10% increase in plant dry biomass. And because there was no changes in leaf hydraulic conductance and high increase in biomass, we see increase in whole plant water use efficiency. So whole plant water use efficiency is the ratio of amount of dry biomass and total amount of water lost by plant. So these numbers, these numbers looks like very small in terms on paper, like around 10% and 7%. However, on the big scale in the field condition, they accumulate big numbers. For example, 7% increase in water use efficiency is can be considered as very high improvement in crop, because a plant loses a lot of water to gain carbon dioxide or to gain yield or biomass. So these studies confirm that thin mesophyll cell wall increased biomass and water use efficiency in rice. So with that, I would like to summarize the today's talk that using natural diversity, we were able to identify various leaf traits to manipulate or to increase whole plant water use efficiency in crops. We validated that thin mesophyll cell wall increased biomass and water use efficiency in rice. In this way, we can use fundamental knowledge of plant physiology to identify traits of interest and use to improve leaf and whole plant productivity and water use efficiency, and develop climate resilient crop for next to solve the global food security issues in changing climate. With that, I'm happy to take some questions.
Thank you, Dr. Pawar. Now the session is open for discussion.
Request all the participants to raise their question. You can unmute yourself or can write question in the chat box, or the student or the participant who are present here can raise their hand if they have any question.
Dr. Pawar, this
Yes, sir.
thin mesophyll cell wall, it has shown the improved mesophyll conduction to the CO2, and how much productivity got increased in such cases?
So in case of rice, the productivity we are expressing in terms of whole plant biomass, not in terms of grain yield. So in terms of whole plant biomass, the productivity was increased by around 9 to 10%.
So it is done only in the rice. Now it can be available for the other crops also, because day by day this global warming problems are continuously increasing, and we are facing the problems of depletion in the crop productivity as well as the various abiotic factors. They are basically focusing or they are targeting the productivity. So in such conditions, this research which is carried out by Dr. Balasaheb Solana sir and this group, it has the solution to the global warming. Okay. So I am very thankful to Dr. Balasaheb for delivering very nice talk on exploring and utilizing the plant ecophysiological traits in improving the crop productivity and water efficiency. On behalf of Anna Sahib Agri College, I am very much thankful to you, Dr. Balasaheb. Thank you.
Thank you, Vinayak sir, for giving me opportunity to share my research. I also thank you to organizer for giving me stage for 15 minutes here, and for you have good wishes for conducting the conference ahead. Thank you.
Thank you. Now, from myself, I would like to thank the organizers, the convener, the principal of the college for inviting me and giving me the opportunity to chair the session on this two talk in one day international conference on exploring the innovations and life sciences, which is organized by the Department of Botany and Zoology. I am thankful to you all. Thank you.
So now we are proceeding towards our third session of the international conference. Now I would like to request Dr. R.J. Jamadar, Head Department of Zoology, ACS College, Narigaon, to chair this session. Okay. Instead of the R.J. Jamadar, the Dr. Kiran Kumar sir is here. I would like to request you to chair the session, introduce the speaker. Over to you, sir. Kiran Kumar sir.
So, friends, you have listened very interesting lectures from foreign speakers or resource person. Basically, Gunawant Patil, a very interesting topic that related to biotic and abiotic stress resistant and total genomics. Where we are lacking, we do anatomy, we do physiology, biochemistry, but we lack at genomic level. So now the another speaker for this session is Dr. B.B. Vaikar. He is Dean and Professor, Head Department of Zoology, Dr. Babasaheb Ambedkar University, Chhatrapati Sambhajinagar. He completed his education up to PhD and having very vast experience of teaching and research, more than 34 years, and he is a good research worker. He has been a recognized guide for PhD in zoology, and under his guidance, about 12 PhD students have completed the degree. This is great contribution by Dr. B.B. Vaikar, I think. Then he completed many research projects and he has contributed a lot of research papers and published literature in the form of books. I think the list says that he has published more than 44 international papers. So research paper publishing in reputed journal having some impact factor is important, and he has done that very, very nicely. He has organized many workshops and experiments, and we are very fortunate to have Dr. B.B. Vaikar here. His topic of today's lecture is apiculture and its economic importance. So very applied topic. I welcome Dr. B.B. Vaikar and request him to go on your presentation. Thank you. Dr. Vaikar, you are able to join, or is there any problem? Yes, Vaikar sir. Is there any internet problem or what has happened? Dr. Vaikar, no response from you. What has happened? Please, please. Dr. Vaikar, are you able to hear me? So I think there is some problem for Dr. Vaikar to be online. So we can shift to other, I think. No, we are just waiting for Dr. Vaikar as he comes online. So I will just talk about apiculture. Nowadays, as he comes online, nowadays apiculture is the most important business in agriculture. Whatever productivity or food we are getting because of honeybees, they are the best pollinating agents in the nature. If honeybees are removed from the world, you will not a grain. You will be removed from the world. So both things are very important. And the honeybees, insect pollination, because without that we cannot go forward. Many times you may be knowing polyhouses and greenhouse cultivation. The tomatoes and Simla mirch cultivated in greenhouse. But honeybees cannot enter into greenhouse and they cannot pollinate. People have to go for artificial pollination. Unless and until they do artificial pollination, there will not be a single fruit. I think Vaikar is. Vaikar sir, are you joining? Yes, sir. Please give response. So, if you go for every time artificial pollination, agriculture ची निम्मे कष्ट आणि निम्मे पैसा या आर्टिफिशियल पॉलिनेशन मध्ये जाईल. सनफ्लावर मध्ये हनी बीज गेल्या नाही तर त्याला एकही बी निर्माण होत नाही. म्हणून शेतकऱ्यांनी त्याला आर्टिफिशियल पॉलिनेशन एक असा हाताने संपूर्ण त्या फुलावर हात फिरवतात आणि आर्टिफिशियल पॉलिनेशन करतात. निसर्गामध्ये पॉलिनेशन हा फार मोठा भाग आहे. इन्सेक्ट पॉलिनेशनला एक गोष्ट महत्वाची असते, अट्रॅक्शन ऑफ द पेटल्स. 3 गोष्टीशिवाय इन्सेक्ट तिकडे जात नाही. त्यामुळे सगळ्या प्लांटनी स्वतःहून काही ऍडजस्टमेंट केलेली आहे. ब्राईट कलर्ड पेटल. दुसरं स्पायनी और स्टिकी पोलन ग्रेन्स. आणि तिसरं दे हॅव व्हेरी स्वीट स्मेल. इज जॉइनिंग? या सर, स्वागत है आपका. प्लीज ज्वाइन.
प्रेजेंटेशन ओपन करो.
Okay. Now we will listen Dr. B.B. Vaikar on the topic apiculture and its economic importance. Thank you.
Screen दिसते? Hello? Hello?
You are audible.
Hello?
Hello?
I am audible.
Yes, we can see your screen and you are audible.
Hello?
Hello?
आवाज येतोय?
Yes, sir, we can hear you and even we can see your screen.
Okay, okay.
हॅलो वायकर सर, आवाज येतोय? हॅलो वायकर सर.
Yes. Can I start my lecture?
Yes sir, you may.
Okay. Firstly, I congratulate the principal of this college, Manchar College, and the organizer. I also very much thankful to the principal of this college for inviting me to deliver a lecture in the conference. Today's my topic is sustainable agricultural development through beekeeping. Know that as a nation, we are facing the problem of population growth. Population is increasing, snowfall rate, and therefore there is a challenge to increase agricultural production by quality and quantity. Presently, Indian population is 121.9 crores, and by 2030, it will be 140 crores, and there is a burden on the farmer to increase agricultural production by quality and quantity. Another challenge is to provide the employment. Among the 140 crores of population, 35 to 40 crores will be the youth in the age group of 20 to 40, and therefore to provide the employment is the challenge. Another challenge, third challenge is a decline in agricultural production by quality and quantity. There are many reasons, but out of these many reasons, two are the main. One is a decline in soil fertility and decline of population of pollinators. The fourth challenge is stagnant resources. As the population is increasing, and therefore per person per hectare land is reduced, shortage of water, and therefore farmer have a challenge to increase agricultural production in minimum land and minimum water. The fifth challenge is to produce toxic-free food, because to increase the agricultural production, we are increasing tremendous amount of pesticides, and because ultimately our food chain become poison. And we know that according to the Swaminathan report, we have accepted, and the first green revolution is happened, and that is successful in mostly in Haryana and Punjab. To increase the agricultural production in the country, these two states using highest quantity of pesticides, and therefore the whole food chain of that region is become poisoned, and number of cancer patients in this area is increasing. And we all of are aware that the one train is running from Hisaner to Bikaner, because in the Bikaner there is the hospital which provides best facility and treatment to the cancer patient. And in that train, the most of the cancer patient, and therefore popularly this train is called as a cancer train. In Maharashtra, we take the example of Maharashtra. In Kolhapur district, Sirola Taluka is there. And in that taluka is known for vegetable growing taluka. They provides the vegetable to the Bombay and Pune city. And to increase their agricultural produce, they using tremendous amount of pesticides and fertilizers. And in a single Sirola Taluka, there 18,000 cancer patients are reported. And therefore, to handle the above mentioned problem, we need eco-friendly technology. We know we all are very much aware that beekeeping is eco-friendly technology. Beekeeping helps to fight against above mentioned problems. Though we are aware that honeybee simply collect the nectar and pollen from the plant and convert that nectar into the honey. And we collect million tons of honey from that source. If beekeeping is not practiced, it is actual national loss. Bee provides free pollination services, and due to the bee pollination, crop production is increased by quality and quantity. That is the important aspect. From the recent data, it is evident that pollination by bee, the agricultural production is increased by an average 40 to 50%, and in general, a plant specific from 40 to 160%. That depends upon the crop species, whether they are cross-pollinated or self-pollinated. Beekeeping is a side business, provide supplementary or a major income to the people of rural peoples. According to the National Bee Board, income from the 100 bee colonies is around 3 lakh per year. This beekeeping for whom? Beekeeping for rural people, poor farmers, landless labor, rural farms scattered over hills, forests, and tribal peoples, and there is no boundary for age and sex. Anyone can take up this activity. You know that beekeeping why? Beekeeping to take the primary benefit of primary role of honeybee. That is, primary role of honeybee is pollination. Beekeeping for pollination, not for honey. After successful pollination, we can go for collection of other 6 bee products like honey, wax, royal jelly. We know that the price of 1 kg of royal jelly is 20,000. The third product is pollen. Fifth product is bee venom. The price of 1 gram of bee venom is 12,000 to 18,000. That depends upon the purity of bee venom. And sixth product is propolis. And therefore, my suggestion is, instead of going for the keeping the honey for keeping the honeybee for collection of honey, we go for benefit of primary role of honeybee is pollination services, because the number of other countries like Israel, European countries, China, Japan, and recently India, we are aware that due to the bee pollination, agricultural production is tremendously increased. According to the agriculture scientists, they reported that it is well established that beekeeping increases agro production 15 to 20 times more than the value of honey and wax they produce. And then we go, therefore my suggestion is to take the benefit of honeybee role, primary role as a pollinator. In India, there are 5 species, actually there are 4, but now we are calling 5 species of honeybees. That is, first one is Apis dorsata. In Marathi, it is called as Agya Madhmasha. Second one is Apis serena indica. In Marathi, it is called as Sateri Madhmashi. And third one is, it is not Indian species. It is Apis mellifera. It is exotic species, but this species is widely used throughout the world for beekeeping practice, because this bee species is a very excellent pollinator, and from this species we can obtain that 6 product very easily. And the 4th species is Floria. This species is also called as garden bee, smallest bee, but honey of this species have a more medicinal value. And the 5th species is Melipona, or it is also called as stingless bee. And this bee is its characteristic is this bee we can withstand with the high temperature also and in advanced environmental conditions. And recently, rightly, this season is mango season, and this stingless bee or this Melipona species is the best pollinator for the mango plants. You know that this is the photograph of giant honeybee species, it is dorsata. This species is very ferocious, but this dorsata is an excellent pollinator. See, single bee visit more than 7,000 flowers in a day, and therefore this species is very excellent in regard to the pollination, to provide the pollination services. Another species is Apis serena indica. This species is used in beekeeping practice, and this species is mild, easy to handle, and in beekeeping practice, the species is predominantly used in India. Third species is Apis mellifera, and we know that this species widely used in the whole world for beekeeping practice, and from this species we can obtain all the 6B product. And Apis mellifera, I want to focus another point. In the bee hive, there are 3 castes of bees. One is worker, second one is queen, and third one is drone. These worker and queen are female. This is also the picture of queen bee. You know that need of the bee pollination from last 10 to 15 years, the focus of agriculture is slowly shifting from traditional cultivation system to high value horticulture crops, farming system such as fruits and vegetables that poses new challenges for improving crop productivity by quality and quantity. Many horticultural crops are actually self-sterile and requires cross-pollination. Logically, the increase in cultivation of cross-pollinated crops will also increase the need of pollinators. We know that we are very aware that there are 2 well-known methods for improving the productivity of the crops. One is use of agricultural inputs such as the use of quality seeds, planting material, and good practices like timely irrigation, organic and inorganic fertilizers, and chemical pesticides to increase the agricultural yield. And the second method is that it includes the use of biotechnological technique such as manipulating the photosynthesis and biological nitrogen fixation, etc. These conventional techniques ensure healthy growth of crop plant, but that work in certain limits only. We know that among the several, and therefore to increase because of input of that fertilizers, use timely irrigation, and all these things, the vegetative growth of a crop may take place. There are abundant flowers are there, but due to the lack of pollinators, the agricultural production is started to decline by quality and quantity. The farmers are facing the problem of decline of pollinators. As to kill the pest, they extensively use pesticides, and ultimately there is no choice due to the excessive use of pesticides. They kill all the pollinators, most of the pollinators, and if there is no pollinator, ultimately the agricultural production is declined by quality and quantity. And therefore, to increase the agricultural production, honeybee is the best option as compared to the other pollinators. There are so many reasons why the honeybee is the best pollinator. The first one is honeybee exclusively visit many flowers of the same species during the single trip, and they promote a natural habitat. Single honeybee visit 7,000 flowers in a day, and in a single colony there are 25 to 30,000 honeybees. We multiply 30,000 into the 7,000 and calculate how much service is provided by single bee colony to the farmers, pollination services to the farmers. Another characteristic of bee is that the body of a honeybee is hairy, covered by the hairs, and hairs are branched and they carry electrostatic charge. When we visit one flower to another flower for the collection of nectar and pollen grains entangle very easily. We are aware that the honeybee has 3 pairs of legs, and on the metathoracic legs there is a pollen basket, and on the another legs there is a pollen. In the mesothoracic legs there is a pollen brush. By using the pollen brush, we collect all attached pollen grains and store into the pollen basket. As this pollen basket is open and we visit one flower to another flower to collect more pollen and nectar, there is a good chance for pollination. And regarding to the potential of long working hours, bees start her duty early in the morning and stop her duty late in the evening. Ultimately, that also depends on the environmental condition, including the temperature. One major difference I want to notice to bring the notice to the all, that is, in the honeybee colony there is a queen. As and when the environmental condition is suitable, flowering is abundant, there is an abundant supply of nectar and pollen, queen bee lay more than 1,000 to 1,500 eggs every day, and therefore it is called as egg laying machine. And as she lays the so tremendous amount of eggs per day, ultimately the colony becomes more strong. If colony is strong, means there are number of workers are there. These workers provide the pollination services, they pollinate the crops. This honeybee has a nature is adaptability even to the adverse condition. And another thing is that the beekeeping is practiced rare and cultured into the box, and therefore these boxes are easy to move from one place to the another place, even one state to another state also. The another thing is the bee is top pollinator. Bee is top pollinator, and therefore she provides excellent pollination services. Another, the approximately 73% of world cultivated crops are pollinated by the bees. 1/3 of human diet is derived directly or indirectly from the bee pollination services. And in India, there are 12 crops that depends on the pollination on the bees. We aware that the well-pollinated flowers contains more seeds. These seeds are bigger and better shaped, and that have 100% germination potential. In case of fruits, the fruits are become bigger and better shaped. Quality is very excellent. These fruits are juicy, and therefore, due to the bee pollination, agricultural production is increased like that way by quality and quantity. I observed in my region, I personally visited so many things and discussed the issues to the different farmers from the Paitan, Kannada and other regions. and I'm aware that in spite of use of improved variety of seeds, chemical fertilizers, water, and insecticides, the first green revolution failed to increase the agricultural production over a sustainable period because of high decline of pollinating bees and other pollinators also due to the heavy use of pesticides. The use of extensive pesticides have controlled the pest to some extent, but that has increased the cost of crops due to the poor pollination by the very less available bees. We know that in Maharashtra or in India, nearly 75 to 80% of Indian populations are farmers by profession, and for them the cost of agriculture is they're facing the problem like high input and low output, and therefore they committed for the suicide. And therefore, I also made interview so many farmers, and I have decided and observed that in my region, particularly Kannad, Silod talukas of Aurangabad district, this area is known for onion seed growing area. This area is also known for pomegranate and sweet lime cultivating area. I made the interview, and they reported that due to the decline of bee population, the agriculture production is declined very fast, and even the quality of seeds of onion. And therefore, I have made a proposal submitted to the DBT and organize more than 55 awareness and training camp on the beekeeping. Total 3,000 farmers, student, and faculty, teaching faculty has attended that workshop awareness program, and among them 300 farmers were selected for scientific beekeeping. And after providing a scientific beekeeping training to the farmers, I have distributed colony to the farmers, and I distributed colony to the farmers. And one thing I want to mention, when initially I have distributed this colony to the farmers, I observed so many things, and I have developed so many models to enhance the pollination and to protect the bee from the use of pesticides. There are so many models, but because of shortage of time, I cannot explain in more details. But one example regarding the onion seeds, I want to explain. In Aurangabad district, Kannad and Silod taluka is known for onion seed growing farmers, and farmers facing the problem of decline of pollinator population, which affect the quality of seeds. Onion seed growers are guided to cultivate 2 to 3% sesame, mustard, sunflower, maize. After the 2 to 3 weeks of onion cultivation, to maintain the flowering time and attract the pollinator, and also suggested to keep the colonies of Apis cerana indica and not the mellifera. Number of farmers using the mellifera species, and my suggestion is to pollinate the onion, use Apis cerana species only. This is the Indian species, and she is well adapted to the condition. I have already mentioned that I have distributed colonies, more than 251 colonies to the trained peoples, and these peoples use this colony to pollinate the crops. And I have collected made the survey and collected the feedback from the farmers, and regarding the 18 different plant crops, and report is that due to in pomegranate 53.49% is increase, sweet lime 71.1% increase due to the bee pollination services, Onion seeds, the 56.80% increase. Pigeon pea 64.80% increase. Means due to the bee pollination, the agriculture production is increased by quality and quantity, and these news are published in so many newspapers. We know that we see the quality of that pomegranate. In my experiment, there were 27 farmers who maintained bee colonies in their pomegranate orchards, and a total 58 acres and 20 one bee box was maintained per hectare of a pomegranate orchard. Without bee boxes, in that area, the production is 410.8 tons, and after introducing of the bee boxes, the production is increased 722.50 tons. Means in terms of percentage, the production is increased 53.49%. And also the tools observed as these news are published in various newspapers. The many scientists and agricultural experts, they visited my experimental plot. This one is the Joshi sir. They come from the Uttarakhand to visit my experimental plots, and there are so many farmers and so many experts. They physically visited the experimental plot. They are located in the different places of Aurangabad district. This one is also the name of farmer is Sheke, and see the result of bee pollination. Due to the bee pollination, they reduce they enhance the setting rate. They reduce the dropping of flowers, and at a time all flowers get pollinated, and farmers stop with the use of pesticides, because as they using the pesticides, we get killed. Therefore, I have suggested to farmers to stop the use of pesticides and keep the bee boxes to pollinate that pomegranate crops, and we see the excellent result. All farmer accepted my suggestions and see the result. These are all photographs are my original photographs. Many reporters are also actually visited the field. They physically observed all the plots. They are located in various plots. These are some plots of pomegranate. This is regarding the sweet lime. We know that in sweet lime also the production is increased near about 71%, and the scientists from the Indore MP, they are visited that study site of my study site. The pollination of onion plants. We know that due to the pollination, the production of onion seed is increased by the 56.80% due to the bee pollination services, and the seeds have the production of the seed quality is very excellent, and they have a 100% germination potential. And another is guava. We know that guava, the 44.80% increase in the yield is observed. Regarding the lemon, we observed that in the lemon, 64.29% is increase in the production. In case of pigeon pea, the production is increased by 64.82%. In case of soybean, we observed the 73.80% increase in the production by quality and quantity. Regarding the green gram, the agriculture production is increased by 43.70%, and in case of mustard, it is increased by the 85.48%. In case of cotton, the production is increased by the 38.80%. In case of per millet, the production is increased by 66.29%. And in sunflower, we got a very excellent result. Even I have tested regarding the result of bees pollination in case of labellum bean legumes, and we observed a very excellent result, that is the increase in the 50% in the yield, and the quality of is very excellent. Regarding the apple bear, we observed a 62.50% rise in the production after placing the bee boxes. Regarding the ajwain, the production is increased by 50%. Regarding the mango, the production is increased by 39.80%. And in bitter gourd, we got the very excellent result. Regarding this, I want to share a very big story. But result are very excellent in the that is say net cropping pattern system, and even in the polyhouses also. And for polyhouse, we always recommend the stingless beans. Regarding the moringa, we observe very excellent result. Regarding the moringa, I observed 50.47% rise in the drumstick yield, and the quality of that drumstick is very excellent. Regarding the brinjal, we observe excellent increase in the production, and therefore there are so many reports are there. Due to the bee pollination, agricultural production is increased by quality and quantity. Therefore, I recommend and suggest all the colleges to promote the beekeeping practice among the student farmers, nearby farmers, and also create awareness. And my experimental result are published in stories are published in so many newspapers. These are some highlights of the newspaper. I want to bring to the notice to the all one notation which is seen by the Einstein: no more bees, no more pollination, no more plants, no more animals, no more man. Therefore, save the honeybee. Thank you, sir, for giving me opportunity to deliver a lecture in your conference. Thank you, sir.
Thank you, sir, for a very, very illustrated account of your research. It is not only technical research, but it has practical applications. You have made compulsory a visit to different fruits, vegetables. We have a nice field visit, and we saw different type of agriculture. Your study is really appreciable and wonderful contribution to the agriculture. We are there because of agriculture. Agriculture is the backbone of Indian economy, and India is the agriculture country. So your presentation was highly, highly appreciable. So thank you very much for nice presentation. Now let us have a quick one or two questions if there. Yes. Any simple question from you, because it's very important apiculture subject. Woman empowerment is there in this subject. yeah. I'm seeing a lot of it.
Hassan.
So I think there are no questions.
Any questions?
Pardon?
Sirji, I will just coming to concluding. Students have fully understood, and I think they have no any queries or questions. It was so nice explanation, no doubt of questions and anything else. So agriculture point of view, you have given very, very authentic information, not only authentic information, but practical applications. The late Prime Minister Pandit Jawaharlal Nehru once said that I am quoting, everything can wait. लग्न बालपण सगळं थांबू शकतं. everything can wait, but not agriculture. So this is what you have given the message, how agriculture is important. Thank you very much. Thank you.
सर सदाशिव बोलबट माझे मित्र आहेत. त्यांनी हा चान्स मला दिला आणि त्यांच्या रिक्वेस्ट मुळे आणि त्यांचेही आभारी आहे.
थँक्यू सर, थँक्यू.
Thank you very much, sir. Before moving on to the next session, I would like to welcome on behalf of Annasai Bhavte College to Dr. Ravi Shirsagar sir, BOS member, SPPU Pune, Vice Principal, Modern College, Ganeshkhind, Professor Mr. Birade, Head, Department of Zoology, HRN College, Khed, Professor Dr. Gurve, Head, Department of Zoology, BJ College, Ade. I request Dr. S.N. Bolbert sir to welcome Dr. Ravi Shirsagar sir by presenting a floral bouquet. I request Bolbert sir to welcome Dr. Gurve sir from BJ College, Ade. I again request Bolbert sir to welcome Professor Mr. Birade sir, Head Department of Zoology, HRN College, Khed. Thank you very much, sir. For next session, I take this opportunity to request Dr. Ravi Shirsagar sir to chair the session. Dr. Ravi Shirsagar sir.
Good afternoon, all of you. It is really a proud moment for the Annasai Bhavte College because they have organized one beautiful international conference on exploring the innovations. And now this is the time for innovate something. You know, the three I's are very important. The one is imagination, another is invention, and another is innovations. So those who are going to invent something, he will be a Nobel laureate. But there are something in our mind that we can bring some changes. That is the innovations. And if you are trying to imagine, so imagination, the Albert Einstein has rightly stated, the imagination is the first step for invention and the innovation. So my dear student, it is the time that you should imagine something and then you should go for innovation and the invention. And for this wonderful international conference, we have the next eminent resource person from South Korea, Mrs. Gitanjali Kanade. Okay. The Mrs. Gitanjali Kanade, she has completed Master of Science in Biology from Illinois Institute of Technology, Chicago, USA. She is expertise in the field of biological science, instrumentation, microbiology, and the molecular biology. She has presented various papers in national and international conference and various summits. The madam has worked as a research assistant at IIT Chicago. She has also worked as a research technician at University of Chicago. She also worked as a quality control personnel at Glenmark Pharmaceutical. She has also worked as an assistant professor at KPP College, Washington. And presently, she is working as a researcher at Suwon University, Suwon, South Korea. So we have a really very wonderful expertise from the international repute. And I must request Ms. Gitanjali Kanade to shower a few words of blessings and the shower a few words of innovations which has been carried out throughout the globe. So I must welcome Gitanjali Kanade in this international conference of exploring the innovations in life sciences. So I request Gitanjali ma'am to kindly proceed with her session. Thank you.
Thank you very much, sir. Thank you for your kind words of introduction. Hello, everyone. Am I audible?
Yeah, ma'am, you are audible.
Okay, then I'll share my screen. Okay. I'll start with my session. First of all, thank you very much on behalf of Raya Shikshan Sanstha's Anna Sahib Avte College for inviting me as a speaker. And my specialization, as sir mentioned, is in biological sciences. So my today's topic is also about talking about exploring the anti-diabetic potential of corsitaglitrin through dual inhibition of PTPN6 and PTPN9. So I'll try to explain this topic as we go along this presentation. The top contents of my presentation are going to include introduction mainly to diabetes, potential targets for diabetes, experimental setup that we have in our lab, the results and conclusion. As we all know that diabetes has become a global pandemic, and over 537 million adults aged from 20 to 79 worldwide are currently affected by this disease, which counts for around 10.5% of the total population of the world. And as per the Indian Federation of Diabetes, this number is going to rise up to 783 million people by year 2045. So diabetes is basically a condition in which you will observe a high blood glucose levels in the serum and high blood glucose levels. And in that case, the causes of type 2 diabetes are many. As we all know that diabetes is recognized as high blood sugar, but there are two types of diabetes. One is type 1 diabetes and another one is type 2 diabetes. In our research, we mainly focused on type 2 diabetes. And as per this slide, you can see the causes of type 2 diabetes includes high blood pressure, pancreatic disease, obesity, age, genetic predisposition, etc. The symptoms of type 2 diabetes would include increased thirst, unexplained weight loss, slow healing, dry skin, frequent urination, etc. So if you see at the molecular level, diabetes is also known as hyperglycemia, that is high amount of glucose in your blood. And it is a complex endocrine and metabolic disorder. And then interaction between several genetic and environmental factors result in heterogeneous and progressive disorder with variable degrees of insulin resistance and pancreatic B cells dysfunction. So in my presentation, I have just a minute. Yeah. I have focused mainly on skeletal cells over here because my research mainly focused on skeletal muscles and the effect of diabetes on skeletal muscles. So in case of type 2 diabetes, you can see the skeletal muscles become insulin resistant. They are metabolically inflexible and they have increased fat storage, and which can also lead to atrophy. Similarly, other organs, pancreas, kidney, cardiovascular system, brain, bone, skin, can also get affected in some other way due to type 2 diabetes. With respect to identifying the targets for treating the type 2 diabetes, our lab mainly focuses on protein tyrosine phosphatase, and these are the signaling proteins which control important and wide variety of cellular responses such as growth, differentiation, migration, survival, and apoptosis. Hence, dysregulation of this protein tyrosine phosphatase is a major cause of human diseases that include cancers, diabetes, autoimmune disorders, and neurological diseases. So basically, protein tyrosine kinases and protein tyrosine phosphatase, they work hand in hand to make sure that cellular function is taken care nicely. But in case the upregulation or dysregulation of protein tyrosine phosphatase, this balance will be disturbed and it can lead to many other diseases. So our lab's main idea was to identify the natural products which can target this protein tyrosine phosphatase and make them as a potential anti-diabetic drugs. So as we all know that there are many treatments that are available in market for treating the diabetes, which include, most of the treatment includes synthetic drugs or insulin injections, but all these synthetic treatments, they have particular side effects with respect to kidney failure, with respect to cardiovascular diseases. Hence, considering natural drugs for the treatment of type 2 diabetes is the ideal treatment option. Hence, in our lab, we first, in order to tackle that problem and also to identify the drugs which can potentially inhibit the protein tyrosine phosphatase, which are the ideal target for diabetes, we first clone 100 PTPs, 100 protein tyrosine phosphatase into E. coli bacteria and created a PTP library. And then all the PTPs were then overexpressed in E. coli. The protein was extracted and purified with the help of liquid chromatography. And after that, these proteins were utilized to conduct the enzyme kinetics assays. And in this enzyme kinetics assay, we are trying to identify the natural drugs which can inhibit the catalytic activity of these proteins. And once we identify the hit compound, we further perform the cell-based assay, which can further validate our study of inhibition of these PTPs by natural products. So as I told you earlier, we have a library of around 1,033 natural products in our lab, and all these drugs were used in enzyme kinetic analysis to see if they can inhibit the potential PTPs which are involved in the diabetes. And we identified the hit compounds after the screening, and these hit compounds were there further validated for anti-diabetic, anti-obesity, and anti-cancer activity. In this similar approach, I proceeded with my study where I targeted two PTPs. One is PTPN6 and PTPN9 with a drug called quercetabgetrin. And quercetabgetrin, as you can see over here, is a flavonoid derivative, and it is obtained from the extraction of marigold plant. So basically, this flavonoid derivative can inhibit the PTPN6 and PTPN9 and ultimately increase the phosphorylation of AMPK and AKT. Increase in the phosphorylation of AMPK and AKT can further increase the glucose uptake in the skeletal muscle was our hypothesis. So AMPK and AKT are also two important crucial downstream proteins which are seen in the anti-diabetic action. They're highly involved in anti-diabetic signaling pathways, and upregulation of these two proteins is related with GLUT4 translocation, and these the GLUT4 receptors are involved in increased glucose uptake. So in order to validate our study first, we made a simulation model, and in the simulation model, we did the docking study. It's like a bioinformatic approach. And this is PTPN6 protein, and this is PTPN9 protein. And these are their catalytic sites, as you can see. And this is our drug quercitagetrine. So after doing this simulation model, we understood that the drug quercitagetrine can very nicely fit into the catalytic pocket of PTPN6 and PTPN9, and hence can inhibit the catalytic activity of these proteins. And if we achieve the inhibition of catalytic activity of this protein, we can ultimately get the anti-diabetic effect. Further, to validate this, we also performed many enzyme kinetic analysis. And in this, we first started with protein isolation and protein overexpression, isolation, and purification. And that protein was further used for identify the inhibitor concentration using quercetagetrine drug, and quercetagetrine was shown to inhibit the catalytic activity of PTPN6 and PTPN9 at a IC50 value of almost 1 micromole and 1.7 micromole. So this was a good data which showed us that quercetagetrine has a potential to inhibit the enzyme kinetic activity of PTPN6 and PTPN9. So it can be anticipated as anti-diabetic drug. So with this approach, we further proceeded with the cell-based assay. And all the cell-based assays were conducted in C2C12 myoblast cells. They are also called muscle cells. So whenever we perform the experiments using these muscle cells, we first had to differentiate them and make them a mature muscle cells and use them for our assays. So in order to use quercetagetrine as a potential drug for understanding the effect of that drug on muscle cells, first we check the cytotoxicity of this drug. And as you can see, at various concentration, quercetagetrine did not show any cytotoxicity, which confirmed that it is a safe drug to be used for cell-based assays. Then after that, we checked the glucose uptake analysis with the help of 2-NBTG, which is a fluorescent glucose molecule. So in this case also, we treated the muscle cells with various concentration of quercetagetrine and analyzed if there is any increase in the glucose uptake in these muscle cells. And quercetagetrine alone was also able to increase the glucose uptake in muscle cells. Further, we also experimented that if it has any synergistic effect in glucose uptake when given with insulin. And as you can see in this graph, as we give the quercetagetrine at various concentration in the presence of insulin, it also increased the glucose uptake. So here we were confirmed that, oh yes, quercetagetrine is able to have an effect of increased glucose uptake in muscle cells. Then we dig deep into understanding what is the molecular pathway it is following to increase the glucose uptake. So we analyzed the most important protein which is highly involved in anti-diabetic pathway is AMPK. And we understood that when we treat our muscle cells, matured muscle cells with various concentration of quercetagetrine, we can see a dose-dependent increase in phosphorylation of AMPK. So we were thinking of a point that our drug quercetagetrine is able to inhibit PTPN6 and PTPN9, which is increasing the phosphorylation of AMPK and ultimately leading to the glucose uptake in the muscle cells. Further, we also explored another most important regulator protein, which is AKT. So phosphorylation of AKT should also be increased in presence of insulin, which indicates that the more glucose will be taken up by the cells. So when we treated our matured muscle cells with quercetagetrine alone and in the presence of insulin, we found that there was a dose-dependent increase in the phosphorylation of AKT when treated in presence of insulin. So which gave us an idea that, or confirmation that quercetabgetrine has an insulin sensitizing effect. That means in presence of insulin, it will help cells to uptake more and more of glucose. Similarly, when a person is obese, when there is obesity, lot of fat deposition can also lead to a phenomenon called insulin resistance, and that insulin resistance can ultimately decrease the capacity of the cells to take up the glucose. So here in our experiment, we analyzed that uptake of the phosphorylation of AKT was increased in the presence of palmitic acid when treated with our drug and insulin. So palmitic acid was basically used to portray the insulin resistance effect. And after the palmitic acid insulin resistance was developed in our muscle cells, we treated our muscle cells with quercetabgetrine and insulin. And you can see over here, the quercetabgetrine and insulin was able to increase the phosphorylation of AKT when compared to palmitic acid and insulin alone. Hence, from this data, we concluded that phosphorylation of AKT increased,
which is used in different instruments are used.
I'll continue. So here it was observed that quercetabgetrine was able to mitigate the effect of insulin resistance, which was caused by palmitic acid in muscle cells. So as per the previous slide, the quercetabgetrine was able to increase the phosphorylation of AMPK. The quercetabgetrine was able to increase the phosphorylation of AKT, and also it was able to increase the phosphorylation of AKT in presence of palmitic acid. And all these data together concluded that when mature muscle cells were treated with quercetabgetrine in presence of insulin or in absence of insulin, were able to increase the glucose uptake by the muscle cells. Hence, in conclusion, we understood that, first of all, quercetabgetrine was able to target our PTPs, PTPN6 and PTPN9. The targeting of these two PTPs will lead to increase in glucose uptake through increase in the phosphorylation of AMPK and phosphorylation of AKT. And these two effects will also aid into insulin sensitivity, increasing the insulin sensitivity and reducing the insulin resistance. So in totality, as per our hypothesis, we were able to conclude that this extract of flavonoid extract from the marigo plant was able to inhibit our target PTPN6 and PTPN9 and was able to increase the glucose uptake in the skeletal cells. So as per this slide, we were able to achieve the effect of more glucose uptake by the muscle cells, reduce insulin resistance, more flexibility, reduce fat, and no atrophy ultimately. So we concluded that quercetabgetrine can be a potential anti-diabetic drug, and it can be used for further therapeutic analysis. Just to give you an idea, currently I'm a PhD scholar in Sungkyunkwan Kon University. This is our University Suwon campus, and this is the School of Pharmacy. I specifically work in Department of Biopharmaceutical Convergence, which is mainly popular for developing the biologics. And this is the overview of my lab my lab is known as Chemical Biology Lab, and we have two sections in our lab. One is Organic Synthesis Lab, and another one is Biological Tool Lab. And I mainly work in Biological Tool Lab. So both of these labs work hand in hand to make a common lab called Chemical Biology Lab. Our professor is mainly involved in antibody drug conjugate, protein engineering, target identification, molecular imaging, and etc. And just to give you a good picture of my lab, these are my lab mates, and he's my professor, and he's currently the Dean of College of Pharmacy at Sungkyunkwan Kon University. And he has his own company named Aptis, and he's mainly into developing the antibody drug conjugates and patents related to that. So thank you very much. And if there are any questions, I will be happy to take them.
Thank you very much, Gitanjali ma'am, for showering the words of knowledge on the diabetes. Now this session is open for the discussion. So if anybody have questions, please raise your hand and ask the questions. This is really a wonderful topic. The ma'am has given a talk on this.
Yeah, it can be little complicated to understand, but I hope I could give some knowledge.
Ma'am, there is a one question. What differences you find the diabetes ratio in India and the diabetes ratio in abroad? So what do you think? Where the
diabetes ratio in India and where you said, sir?
Yeah, abroad. So like
abroad.
Yeah.
Actually, in India, as if you see this particular graph over here, just a minute, I'll share my screen again. Yeah. So these the one that are highlighted in dark red color are having the most population of diabetes. So currently India come in the range of 6 to 10%. And globally, there are other nations also who have higher percentage of diabetic patients. So, but currently, I feel India is also having a very steep rise in diabetes patient. So we should be more careful. And actually, diabetes is not a disease for which you have to take medication and resolve it. It's a lifestyle disease. So basically, if you maintain your lifestyle and eat healthy, work out or exercise every day, it can be maintained very nicely. So there are no hard rules, and hence that is the reason many researchers are working on finding out the alternatives for diabetes treatment rather than taking synthetic medication. Are there any natural product medications that can be used for diabetes? So basically, I feel it's a lifestyle disease and it can be treated easily if you change your lifestyle.
Yeah. Thank you very much, ma'am. Is there any question? Yeah. Ma'am, there is one question. What is your name? Yeah, Pathan Ashwini. Ma'am, there is one question from Pathan Ashwini.
Oh yes, sure. I will take it.
What is the chemical nature of QG? She's asking.
What is the chemical nature of QG? Oh, the QG is, I feel it's, as I mentioned in my presentation, it is a flavonoid. Actually, I am not a very chemistry person, so I can just tell you that it's a flavonoid, and most of the flavonoids have shown to have a good anti-diabetic activity. So when we did a lot of screening of 1033 drugs, we understood that this is a flavonoid derivative and it can have a potential anti-diabetic effects. Hence, we selected this drug for our study.
Thank you, ma'am.
Thank you.
Thank you very much, ma'am, for your wonderful session. I express my heartfelt gratitude on the behalf of Anna Saheb Bhavte College, Manzar, and the Department of Botany and the Department of Zoology. They have organized a beautiful international conference on exploring the innovations. My dear students, you know the whole world is suffering from the diabetes, and in diabetes you'll find the type 1 diabetes and the type 2 diabetes. In type 1 diabetes, you'll find the pancreas is not working and the insulin will not secrete, and the person is suffering from that diabetes. But in type 2 diabetes, you'll find that the insulin level is low in the body and the sugar level goes on increase in the body. Now it has the time that we should work on this mitigation of this diabetes, and madam has given a wonderful light on this diabetes. So thank you very much once again, ma'am. I express my gratitude on the behalf of all the participants on this college. Thank you very much for this wonderful session.
I would also like to raise a vote of thanks, especially to my friend who invited me for this talk and gave me an opportunity on this beautiful stage to express few insights of my research. And I, as you guys mentioned about me, I was also associated with Rayat Shikshan Sanstha, and it has really given me a lot of opportunity to groom myself. So I wish all the best to all the students. Please explore research and development if you're really interested. There are a lot of options in South Korea as well. You can get 100% scholarship and pursue your master's or PhD degree. And if there are any questions, I will be happy to take it. You can take my number or my my email address from Professor Hemangi, and you can contact me if you have any questions in that regard. Thank you so much.
Thank you very much. Now, I must express my heartfelt gratitude towards Professor Kokre sir, because he has invited me as a chairperson for this session. And I must express my gratitude towards the Department of Botany, Department of Zoology, the principal of this college, and I must express my mentor, Professor Dr. S.B. Patil. He is also joined here as a principal. He himself is a big scientist. So thank you very much for giving me this opportunity to chair the session. Thank you, ma'am. Thank you once again. Thank you all the participants. Have a wonderful time. Thank you.
Now we are proceeding towards the fifth session of our international conference. Now I would like to request Dr. Gulwe, HOD of Department of Zoology, DJ College, RA, to chair the session and introduce our today's speaker. Over to you, sir.
Thank you. Good afternoon, friends. So, next session of this today's international conference is of Dr. Ram Gopal Devdas.
Yes,
I
sir.
am going to introduce Dr. Devdas sir. Dr. Devdas sir is presently working as a principal scientist in National Research Center of Orchids, ICAR, East Sikkim. His research area is genetics and plant breeding. Sir has completed various training courses, germplasm characterization on jhum rice, maize, oil seeds, pulses in Tripura orchids at Sikkim. Sir has also developed crop variety of field pea. Sir has also developed genetic stocks. Sir has obtained various funds in several lakhs for various research projects. Sir has worked as an associate editor for Indian Journal and Applied Orchid Research. Sir is a member of Sikkim Biodiversity Board, and he is also a life member of Indian Society of Genetics and Plant Breeding. So today we have with us this renowned personality in the field of genetics. I once again welcome Dr. Ram Gopal Devdas sir on behalf of Department of Life Science, that is Botany and Zoology, Anna Saheb Aute College Mansur, and all the participants, various delegates of this today's conference. Once again, I welcome you all also, and I request Dr. Devdas sir, please speak towards
our delegates and various participants in the various fields of life sciences. Thank you very much, sir. Please start your presentation.
Yes, sir.
Over to you, sir. Thank you.
Yeah. Sir, is it visible?
Yes,
Hello.
sir. Yes.
Yeah, is it visible? Sir, thank you for this opportunity. And sir, everybody knows about various aspects of inventions and their protection. And fortunately, unfortunately, even though I'm product of ICRISAT from Hyderabad and the University, APAU time, and then subsequently in collaboration with Washington State University, I completed my PhD. But somehow my journey and research was completely devoted to the northeastern side, sir, where I got opportunity to work on various aspects and then very much world crop, that is orchids as well. And then gradually I developed in consonant equal to this Indian acts. And in 2001, there is one act has been passed, the Protection of Plant Varieties and Farmers' Right Act. So I developed certain concept and streamlined some guidelines. And then I got one meritorious award for the farmers, actually. That is Plant Genome Saver Community Award. So I have been, it was awarded in 2017, and nobody else could able to come up with that strategy and able to do it again. But all those things are very much related to the farmers, and that also covers actually plant breeders' rights also, that is intellectual rights also. Apart from that, there is a tail and sui generis mechanism has been developed for IPR aspects. And so those are, I have that good comprehension and then good understanding and recognition. So I would like to highlight this one to the at various forums and this thing, because it is this my expertise genetics and plant breeding has come from applied botany, but I'm not a botanist, sir. So this particular aspect was very much recognized, and then nobody able to break through this particular record. So myself and then Dr. Ram Gopal, and then she's my PhD scholar. She got doctorate also. Rubina Sharpa, on behalf we are presenting, sir. Sir, the network is a little bit slow. I'm just that's why PDF only. I'm just sharing actually. If you see this picture, sir, you see that moon travel was imagination. It's a dream, but it was materialized by involving almost 20,000 scientists and technicians in NASA in Apollo 13 mission. So it was materialized, and sir, no, you might have seen people flying kites. It was there, and then grandmothers' tails were there to landing on moon, but it was done by only few people, sir. That is apart from that NASA and then all endeavors, and that has gone. Van Braun, Professor Van Braun, is a German, a war convict. He was taken and kidnapped by CIA people from US to know the technology. So this is the importance of invention and technologies. If you don't protect those things, whether it is biodiversity or engineering and technological advances, we are nowhere. Whether it is Oppenheimer, also same thing. Otherwise, who will invent this particular atomic bomb and that one? And the same is with that Saturn engines. So many brilliant scientists in US, they failed to develop actually suitable engines take uplift that from that gravitational force and take to the outer space or on the moon. So these are all, I'm just telling, sir, these are all great imagination. It was even when it is Saturn engines were tested, they have taken help from US Army, because that particular vibration and this thing, even buildings will blast and our eardrums will blast. So this is at the same time, if you don't protect, there will be certain person like terrorists also will be there near our surroundings. So one of the quote what I like, no sir, this is all combination of several this thing, all the military generals and then great thinkers and then Armstrong, then who is a veteran MiG-27 operator, and he was taken into this NASA flight mission, and then he was a theoreticist actually, but he was that everything applied part was done by other colleagues and scientists, and then Carl Sagan, and Carl Sagan is one person that he developed. He has taken Voyager 1 and 2 missions to take snaps, and he took one snap from Saturn, and when that that Voyager 1 it is crossing, he took one snap, putting some filters, and this is the spec of this our planet Earth, this thing when it is seen from Saturn, that is planet. And what is our existence, what is our value, this thing. It's only a dust in the whole gigantic university. So with this inspiration, I'm going there, sir. On this planet, we have got very vast diversity. And I have been stationed almost for 15 years in NRC Orchids. Still, I am in Sikkim only. And I would like to help something about Sikkim as well, because this is the one edge of near to Himalayas, actually. You will see vast diversity. It has been done a lot of, I've seen the Maharashtrian researchers and botanists. They are all good botanists and taxonomical botanists. They are doing wonderful research. But really, sir, no, Sikkim is heaven for whether it is philosophers or travelers or botanists, it is a heaven. Same in the case, you see, sir, no, this is all actually Jurassic period evolved this Orchidaceae family, sir. You see the diversity, unimaginable, you will not find anywhere else, because Sikkim is 88% of the land is forest area, sir. It has been protected. Nobody else go inside and then take any collection also, even for research also there are restrictions are there. I'm a member in Sikkim Biodiversity Board also. But I used to ask my even friends and colleagues also, those who want to take that, you take permission from the appropriate authorities, legal authorities, so that you will be protected. Otherwise, that whoever is coming and taking for research purpose or for beautification purpose, it will be under act. So same in the case, sir, that very wonderful thing is that actually there are 7 species are there. They are all actually state flowers of different states. And suppose Nobili orchid is for Sikkim, sir, and as well as lady slipper orchid for Meghalaya. And then that Kopuful Rincostellar retusa for this state flower of Assam. Similarly like that. And then indirectly, Mizoram, not directly, that Vanda is the state flower of this Mizoram. And then coming to the biodiversity, sir, I'm just telling that already is that the Sikkim, that the forest cover is almost above 82%. And how much is left? And 12%. 3% is only for buildings and roads, and then 8% is only devoted for the that agriculture cultivation. And this is the aspect of Sikkim, sir. Most of the area in North Sikkim is not accessible and protected area. It requires underline permit and then permission from DC also to go into that. But otherwise, this is the diversity of this thing. Are we protecting? My question is, sir, we are keeping everything in taxonomically in glasses and herbariums. Are we protecting? People are taking air samples, they are taking soil samples, they are taking water samples to identify the new innovative things embedded in the Himalayan biosphere zones. And these are the wonderful people those explored Sikkim in 1948 to 50s. So J.D. Hooker, he visited this and he documented a lot of diversity. And then this is the zones are actually, it has been taken the border for this China and Tibet actually. It has been banned by our Home Ministry not to display. So one painter has come because there is no restriction on the paintings actually. Photos, there is a restriction there. It was withdrawn with the British's time also. And this is the wonder level documentation. This is the documentation of orchids. It is done by our colleagues and my scholars at NRC Orchid. And this is the amazing diversity we have. And you know, sir, no, Sikkim's the secondary center origin for the maize. And you will not see that kind of various diversity even in Brazil and Peru also. That where the primary center of origin was recorded in South American continent. So this kind of, and then you know, sir, no, we have got colored rice, colored maize, and longest crop size also it is there in the germplasm of Sikkim collections. So next is that, sir, I'm just focusing on that actually. Suppose how many lines we have, like land races and this is there, and then versus how much it is protected. Suppose that plant variety protection status in 2 years back is that. And these are the registered one was there. And then there is very less focus on the indigenous fruits and sugar crops and spices and cucurbits and as well as flowers. So altogether, we are claiming almost thousands of species diversity. At the same time, when it comes to the commercial exploitation and then for production aspect, actually it is very less, very, very less. And if you see, yeah, mostly cereals are more and fiber crops is more because of the demand for the jute fiber and then other cereal crops, including paddy and then maize. So this is very less. And that's what I want to sensitize all these things. And I would like to show that what I have achieved. No civilian award in India is having more than 2 lakhs or 4 lakhs, including Paramveer Chakra. But farmers, that somebody was commenting, everything is first, but farmers, agriculture cannot be neglected. One Professor Sir was telling that is true. And then the highest civilian award for farmers or group of farmers is about 10 lakhs now. That is the value of that protecting biodiversity. Sir. Same in the case, there is one wonderful line, sir, there that is called primitive maize, and that is the kind of teosinte type and a type of rye type or barley type lines are there, and they are very much that corn blight resistant and cold resistant genes are there. That if somebody researcher or botanist or somebody explorer or a foreigner comes, he takes away the plant, we will lost. Unless you claim the protection, it will not protect it. That's what I want to say, sir. And of course, sir, there are certain types are there in the context of global these things, and everything is time bound, sir. If you don't file it, we are nowhere. Because Sikkim large cardamom and then that Sikkim Delhi chili that is called red pepper cherry, that has been GI. So those IC are trying to do package of practices and standardization of regulations for export market, and then fulfilling that organic certification standards, being Sikkim has been declared as organic in 2016. So these are the things we need to standardize these things for export marketing also and certification standards also. And then coming to the now, sir, no. If you see this thing, only one option is left for protection from the botanist side as well as farmer side. For me, a good farmer is a botanist only, because he streamlines and he makes things readily available, and then we got benefited. Even applied breeding also, we got benefited from the whatever output has come from the botanist. Suppose if you see in the legal terms, actually, the enforcement was started from 2003, sir, 2005 notification for several crops. And then these are all the crops that has been already notified. You please see, sir, because none of the people from botany they are not thoroughly aware of this particular guidelines and then notifications. And when it comes to my specialization, yes, I'm a good specialization in cereal crops and legumes and then this thing, but I'm very poor in medicinal and forest and plantations and other cash crops, unutilized vibrant species. So far, what happens, sir? New varieties, this is the output of plant breeders or scientists. It is very less. And then this is for the past developers and scientists who developed the new varieties. Today, we are full sufficient because of this particular incident, and because it is green revolution we achieved because of this act. And for immemorial times and thousands of years, we are sustaining in this continent because of farmers' contribution. There are a lot of varieties are so special. Even you say that example of this basmati, it's a farmer's variety. It's not any scientist developed variety. Nature has given a scope to develop that. So are you protecting that? In which way are protecting? So far, we claim actually so much of biodiversity, but we have got authenticate, legitimate certificates. We got only 5,000 only. And you know that similar to the patent, it is having around 15 to 18 years that validity period. So when it comes to time bounds at this particular thing, I want to throw. Suppose if you want to say protection of any other local lines or farmers varieties or indigenous lines of you, for breeders, for scientists, the time is up, sir. 2020 is the last. Now 4 years is up. This is varieties of common knowledge, means that has been already existing. That is also time is up. Only farmers has an option to file those particular lines. And I came to know that a lot of lines, even from some parts of that Maharashtra also, they filed recently for special rice, black rice and something like that. That time is there up to 2024. The problem with this international laws and then time bound this thing is that after this particular 24 lines, we no longer can claim any legitimate protection over this material. So that's in that sense, actually, botany also coming to me, and even for vegetables, even for other winter vegetables, and then some other perennial cash crops, and then some other forest plants like tree and apple and apricot also. Hello? Yeah, yeah. Is it audible now? Oh my God. Then what I will do, because okay, its network is okay, madam. Is it not audible at all? Okay, because I'll just now it's audible now. Is it audible now? Because 1 minute. Okay, because I will switch off this now. I will just phone. I'm keeping that off. Okay, okay, take it. Okay. So when it comes to the international plant variety protection, no, sir, actually that land races genetically diverse and locally adaptable to traditional farming is also eligible for it. And then contrary to the that conventions, actually Brazil, India, and China developed actually its own system actually where that indigenous lines can be protected. Similarly, now being that organic cultivation is being in popular, actually, there is one committee is formed in the name of Cobra also, and where actually they were again focusing on the organic plant breeding also. This is also in future actually to gain economic future that is very much required. When it comes to orchids, sir, why was so fascinating is that they belongs to this mid-Cretaceous period, sir, around 40 to 80 million years actually. Whatever we see orchids today, they are all actually very primitive taxonomy and then Orchidaceae family, and they represent just some kind of evolutionary stage and status, and they appear like some great big insects. So just I'm not going into much about this. This is also same thing that status of the particular this thing, and then this is the classification as per that this thing is that this particular primitive that was highlighted actually that is kept under CITES Annexure 2, sir, the protected zone. Same with the case that one of the wonderful orchid we have in Sikkim and Meghalaya is that actually lady slip orchid. So one few hybrids also I developed in this and I filed for protection. So this is the kind of I developed some pedigree status from the most famous, the Winston Churchill. So Winston Churchill is basically developed in US by that Royal Horticulture Society, but pedigree comes to actually Indian side. It has been collected here in India, sir, like Insigne, Villosum, and this thing, no? That this is all the indigenous sources. We got it from they got it from India, and so much of exploitation has been done. It has been collected from Professor Hooker onwards. Actually, it has been taken. It has been taken and kept it in good display, and then it is reinstated and it was recognized again only when the 2016 our expected Prime Minister Modi sir visited. Then he named 3 orchids, and everybody got into attention that what are these hybrids? This Cymbidium hybrid, and then Deen Dayal Upadhyaya name, and then Sardar Vallabhbhai Patel, and this is called Narendra Modi. The same is the case, sir. Why I could able to develop some guidelines and make these prospects, and then it is just and not only it is getting a recognition and protected, and I will also commercialize those varieties. So here I just throwing something, sir. Most of the seed companies, if you don't do, other foreign companies will be dominating. Even you know that Maharashtra is a good seed marketing in India, but nowhere stands in the global market. You see that most of the things it has been governed by Bass and Bayer and then Monsanto. These are the dominating seed field. And these are the prospect for the dominating field of the private companies dominating in this field. Suppose if they will be taking your own farmer's line and they will modify that and they will sell it. So this is what will happen in the business and the commodity business of seed actually, even though it is protected. Indian Act can repeal it and then make it available, but still they have got protection issues are there. So for in terms of this plant breeding point of view, sir, and actually whether it is conservation will give protection, that is the meaning of it actually. When it comes to this conservation, preservation, you know it, and if you are keeping for preservation, you are not able to maintain its commercialize it actually. So one of the things that has come for protect is actually GI, geological indicators also it is coming, and then protection of plant varieties and farmers' right also. So there is some controversy there, sir, that we don't do that, what will happen? Suppose recently in the 2012 there is landmark judgment is there, sir. This is $200 billion penalty was imposed on this Monsanto company because they didn't repeal actually after exhaust of 15 years of that legitimacy to use that variety also. They imposed actually a kind of extra premium on the farmers, and then in 2012 that judgment has come. And you might remember this is the same case. It happened with that Land Rover, that company also, where it is the penalty almost for $28 billion on that omission, that what is that non-complying actually that BS6 standards, and like our country is also implemented actually that BS6 norms for actually all vehicles. So that emission should be very nominal actually. The emission of the carbon dioxide and monoxide should be very less, and otherwise it comes under, it attracts and it under act actually. Same is the case with our agricultural commodities and agricultural protection laws also, sir. In brief, these are the laws are there, but where except this GI and then PPFRA, this is under 2001. These one only will give actually brighter opportunities for farmers, and one should botanists also should aware of this thing. So this is all actually developments, sir. Maybe maybe you people know that CBD convention in 1992, it has given into access to plant-based resources. You can also access and you can also give it actually, but under particular terms. And TRIPS agreement has come into 2000 enforced actually because of the United Conventions actually. And after that, we formulated these laws. And we have got only limited time of a 5 year or 15 years time for us for Asiatic countries. And otherwise, the developed nations, they're already having these laws. So this is a broad map of that, related agricultural laws. And a TRIPS agreement tells about this article number 7 actually. This is only for welfare, and ultimately it will promote for technological promotion and transfer of technologies for mutual advantage, and then having balance of rights and obligations. Please, one has to respect this. Same in the case, this has been UPOV has been formulated, but it has been reinvented and then came into the PPV, protection of plant varieties. Same in the case, the TRIPS, what is the liabilities and this thing, it has been highlighted. That means promote that public interest for technological development. At the same time, that international transfer of technology should not affect this thing, the both in balanced manner. So these are the, you may be knowing, sir, this is the standards of IPR, TRIPS number, article number 40. So everybody knows in publication we are doing that, engineering fields we are following that, all the commerce related GI is following that, industrial designs are there for IT sector, for scientists patents and protection of plant varieties, both are there. So, and it comes to internal circuits and then like Pepsi, Coca-Cola, that protected undisclosed of information is there, and then dumping also being controlled. Suppose, sir, I did, because what happens in earlier cases actually, 1966 Act is the most viable thing, so seed standard and then seed certification agencies and the tax and these things are there. But this is also valid for only controlling up to some extent. These are the forms, sir, we have. Suppose any student also, if they will deposit any seeds, NBP, they will get one legitimate number. And then that it will be there in data, but there is no legality. Suppose if you do same thing, same norms, which is very hidden cost, there is no hidden cost, very nominal charges are at 1,000 rupees. Farmers' cost is nil. Under protection of plant varieties, if you make it actually legal, it is giving rights, and then you have 15 years right, and then exclusive rights you have. So these are the certain orchids I developed actually. Very exclusive ones. I can do both licensed application and non-licensed commercialization also I can do. The publication will give a notion and publication only will make documentation only. When it comes to this thing, we will get a certificate. And when it comes to another thing, we will get another certificate. But these are the lines, who will give the certificates? How many times I have to make this? Only wonderful source is that I need to establish. You might be knowing, sir, those who worked especially in the botany. Yes, this is all Cymbidium hybrids only. This is called lady simple orchid. And from this, I developed several clones, so tissue culture and then hybridization programs. So how to protect them? And so there are certain clauses are there in this actually. Even this is very, very simple and then very distant. I'm just telling you that by filing these things, I got my farmers, I did for my farmers, and then highest civilian award for 10 lakhs award has come. Now it is going to be revised. There is gene fund is created and then 15 lakhs and 20 lakhs is coming. So one should try and one must try these things, sir. And these are all actually several steps for getting that particular plant variety protection rights. And this is the approach and schematic representation that test testing will be conducted for 2 years. And then subsequently 13 years rights will be given, sir. And every 5 years we have to renew that actually. Like a patent also we used to do that. And these are the typical type of filing requirements, sir. Actually, we need to attach these things. For farmers varieties, you require department certificates, so that's all. And you very surprisingly, no, sir, you need to, the farmers varieties, the botanist varieties, it is only nil, sir. So that is what advantage we have. And then these are the forms where I filled up, sir, because as an individual breeder we can do, as an application we can do, and a community of farmers can do, and a group of farmers can do, and any other persons also can. Foreign countries also can come because through authorization only they have to come, and otherwise there are certain no permission is there. It is only for only civilians only. So this I did for when I was working in soybeans at Indore. I did for my institutes. There are very popular varieties like JS335 and JS95, and I did for NRC 37. And then apart from that, I did actually several other varieties also. Like I will show, sir. This is all the technical questionnaire. You have to fill up minimum things and existing data only we have to provide. And ultimately you will get, sir, like this is what I got it for Ahilya 4. And I got it for next most popular variety after JS335 in the southern India and then central India. That is called 9560. So that also I framed in this thing. And in terms of revenue, no, sir, actually, it's huge actually. Like whether it is breeder DAC intent or breeder seed intent, it will be most very much encouraging, and it will be protected. And we can claim proudly that this is Jabalpur variety, sir. It is developed by N. Srivastava. But he retired, and then I took that charge and I made it, and it's very glorious to him. He to especially thank the professor like A.K. Srivastava. He told my son, this has been I never expected because the time has changed. We cannot catch up this thing. So this is what I got it, sir, and these are all other things are there, and this is whatever I should say, whether it is botanist rights or breeder rights, and I consider both are same, sir. When it is done, actually, you are holding a patent, sir, and you're helping farmers also to make our sustainability is there. So researcher rights are there. We can register, and then you can. But there is one thing is there, we can do research for other developers variety, but we cannot commercialize it. If you want to commercialize it, we have to take authorization from the breeder. Otherwise, it will be giving complication when it filed to go essential developed varieties in. So this is what in nutshell I want to say, sir. Thank you so much.
Not audible, sir.
Sir, actually, I am a resident.
It's not available from that side.
Is it audible? उनको सुनाई दे रहा है?
Yeah. Now it is audible.
Good afternoon, sir.
Yeah.
Actually, I am a resident of Darjeeling, which is very close to East Sikkim,
Okay.
and the beautiful orchids have really caught up my attention. So my question for you is, in your ideology, which field has been yet unexplored with respect to orchid that would make a really innovative approach in the field of pharmacological life sciences?
Actually, we did actually certain pharmaceutical identification through advanced studies. Actually. Most of that dendrobiums and bulbophyllum and cymbidiums, they are found to have actually anti-cancerous properties. And my specialization actually crop improvement, and that particular aspect I've not gone through. But mostly I focus on hybridization and mutation breeding programs and de novo sequencing those studies, and then filing those gene sequences for NCBI and this thing. But when it's come to pharmaceutical and then those aspects, there are Professor Dr. Nattu is there actually. He is a biochemist and who is dealing with that. And I'm just telling, no, this particular area has been not explored actually because of lack of awareness actually. Because mostly, even you know that Srinivasrao Patil sir visited. He was earlier the governor. He's from Maharashtra. He's from IAS cadre, but he worked as a governor, and he visited several times my farm and then my institute and my lab, and he was so fascinated. And I told sir, this is particular dendrobium species, sir, where they have got anti-cancerous properties. The one of the thing is that whether they will be more or less it looks like a potato plant, potato tubers only, but they've got some form of that condensed stem will be there. They will be reserving that, and then you know that this is epiphytes, and they will not grow on this soil, and then they will again grow on orchids. That is something like a monkey crop, but that particular mechanism, and it has got CAM pathway. Neither it is C3 and C4. All agricultural crops are either C3 or C4. But this particular scam pathway, it breathes and takes synthesis photosynthesis in the night period only. That's why it is all the biochemical properties of the stored products. No? It is so valuable. It is under still studies actually. So this particular thing we started profiling under one project NHMS, and because of Corona period, it was a little bit disrupted. Again, we started and we identified some compounds, and then we are trying to commercialize that also. So that will be having each one crop and each one species will be meticulous and miraculous. And then you know that no, any crop evolved prehistorical times, and then yes, around 30 to 80,000 million years means when the Asiatic component Gondwana thing it got separation. That time it is created, and they are existing all over the world except Antarctica and then polar regions. It is not there. And then you see that this our Sikkim more or less climate almost looks like what is that biosphere zones, no? Like mega biodiversity zones. And then this particular area only is harboring almost the next second highest biodiversity for orchids next to Peru. Peru is having the most richest density, highest density is there in the Peru and then this South American continent. Next, that only Sikkim is having that. So even forest department will not give permission. Lodging professor will never give permission to me because if he tells that yeah, if I give permission, that's right. But I have given to one scientist, so they will be all citizens and private entrepreneurs will come, and everybody is not honest, and they will take away the material. So that is what, and this particular biochemical aspect is done. We are slowly progressing at our institute. Is it okay? Yeah, thank you.
Anybody from audience? Thank you, sir. You have explained very nicely regarding protection of plant varieties and also how to protect orchids.
Yes, orchids.
You have mentioned various central government schemes, various agencies, seed companies, how they are going to protect the crop variety and orchids also. So in our Maharashtra, there is one lady called Rahibai Popri.
Yes, sir.
She is not well educated. She is staying in rural area and she has protected thousands of crop varieties. And for that purpose, Government of India, that is Presidency, got President Award.
Okay.
In last 2 years. So that is the need to protect such and biodiversity. So, sir, your work in this is very nice, and thank you very much for enlightening all of us regarding plant protection, crop protection, and orchids. On behalf of Department of Botany and Zoology, Honorable Anna Sahib Aute College Mansar, I thanking to you for giving this nice talk by accepting the invitation of this today's conference. Thank you very much, sir.
Even in fact, I'm very thankful to
So
Golbert sir,
to
who was
the
coordinating
principal of this college.
Yeah.
Thank you, sir.
Thank you so much. Thank you. Bye.
I am also thankful to Principal Dr. Gaikwad sir, the convener, Dr. Pokre sir, and all the staff of this college for giving me an opportunity. Dr. N.S. Gaikwad, Dr. Golbert sir, Dr. Pokre sir, Dr. Kumawat sir, for giving me an opportunity for chairing this session. Thank you, all the team. And the session is now over. For next announcement will be, yes.
We will have oral and poster presentation after lunch break at 2:30 p.m. Oral presentation will be conducted